Quantum reports. Review. Quantum biology, an update and perspective. Youngchan Kim, Federico Betania 1, Edline M. D'Souza, Darren J. Hayes, Evelyn E. Tineri, Antonio Pantelius, Alejandro Sanchez Pedreno Jimenez, Louis Slope, Michael G. Spencer, Jim Al-Khalili, Gregory S. Engels, Sam Hafe, Suzanne M. Hingley Wilson, Kamalan Jeevaratnam, Alex R. Jones, Daniel R. Katnig, Rebecca Lewis, Marco Sacchi, Nigel S. Scrutton, S. Ravi P. Silverth, and John Joe McFad, Linus O. Johannesson. 1. Introduction. Biology has traditionally been considered to belong to the classical domain, while physics and chemistry are deeply entrenched in quantum mechanics. Yet living systems are fundamentally quantum mechanical, since the dynamics of their molecular, atomic, and subatomic chemical machinery is, like everything else, governed by the law of quantum physics. Does this atomic scale matter in biology? Does life need quantum mechanics? In other. Citation: Kim E. Betania F. D'Souza M. Hayes D.J. Johannesson, Lowe Neri, E.T. Pantelius, A. Sanchez Pedreno Jimenez A. Slocum L. Spencer M.G., and others. Quantum Biology, an update and perspective. Quantum Rep. 2021, 3, 1-48. doi.org/1ang0 3390/.quantum30010,006%. Correspondence. At surrey.az.uk abstract. Understanding the rules of life is one of the most important scientific endeavours and has revolutionised both biology and biotechnology. Remarkable advances in observation techniques allow us to investigate a broad range of complex and dynamic biological processes in which living systems could exploit quantum behaviour to enhance and regulate biological functions. Recent evidence suggests that these non-trivial quantum mechanical effects may play a crucial role in maintaining the non-equilibrium state of biomolecular systems. Quantum biology is the study of such quantum aspects of living systems. In this review, we summarize the latest progress in quantum biology, including the areas of enzyme-catalyzed reactions, photosynthesis, spin-dependent reactions, DNA, fluorescent proteins, and ion channels. Many of these results are expected to be fundamental building blocks towards understanding the rules of life. Publishers note, MDPI stays neutral with regard to jurisdictional claims in published maps and institutional affiliations. Copyright 2021 by the authors. Licensee MDPI, Basel, Switzerland. This article is an open access article distributed under the terms and conditions of the Creative Commons Attribution, CC, by license https creativecommons.org licenses by 4. Quantum Rep. 2021, 3, 1-48. doi.org/103-390/quantum301/quantum free. www.mdpi.com/journal/quantumrep.quantumrep 2021 3. Words. Can quantum mechanics play a fundamental role and have a physiological impact in biology? Most importantly, are there any biological molecules or biomolecular systems that exploit non-trivial quantum mechanical effects to achieve biological advantages that cannot be understood within the boundaries of classical physics? Many of the early pioneers of quantum mechanics thought that the answer to this question was an affirmative yes. 1. As most famously argued by Erwin Schrödinger's highly influential book, What is Life? 2. Yet, until recently, most approaches to investigate quantum effects in biology, like Schrödinger's, were entirely theoretical. This was largely because experimental approaches capable of detecting delicate and fragile quantum effects in biological systems that were dominated by random molecular interactions had not been developed yet. However, at this stage, it is useful to define what we mean by quantum effects in quantum biology. As already pointed out, at its most fundamental level of particles, atoms and molecules, biology, life, like everything in the world, is governed by quantum laws. For example, the structure of electronic orbitals and chemical bonds, what are sometimes referred to as examples of trivial quantum mechanics, are as central to structural biology as they are to chemistry or physics. Yet structural biology is not usually considered to be a branch of quantum biology. What is meant by quantum biology is the involvement of phenomena that are normally confined to the quantum realm of atoms and molecules, such as coherence, tunneling, entanglement, or spin, because their effects are normally cancelled out at the macroscopic level due to decoherence, too, and would thereby be thought highly improbable inside the warm, wet, and disordered environments inside living cells. More importantly, to be non-trivial, such effects must explain or drive a biological process that is likely optimized by evolution. That is, for a quantum effect to be non-trivial, biology must have evolved to exploit it. 3. In contrast, a trivial quantum effect may be detected but just arises from the material being made of atoms, molecules, electrons, etc., all of which are described by quantum mechanics. Given these limitations of quantum biology in its earliest decades, it is not surprising that significant discoveries had to await the development of a range of observation techniques, tools and approaches that enabled the measurement of complex biological molecular dynamics with unprecedented accuracy. Although some of these approaches have been met with strong criticism, 4 to 7, evidence is mounting that non-trivial quantum mechanical effects play a fundamental role in biology. Earlier reviews on quantum biology are available elsewhere, 8 to 13. Here we summarize recent insights into quantum biology, focusing on 6 putative quantum biological phenomena: quantum tunneling and the importance of protein dynamics in enzymatic H-transfer reactions, quantum effects in photosynthesis, magnetic field effects on spin-dependent reactions in biology, candidates and constraints, proton tunneling in DNA, fluorescent protein as a novel model system for quantum biology, and quantum coherence in neuronal ion channels. 2. Quantum tunneling and the importance of protein dynamics in enzymatic H-transfer reactions. 2.1. Introduction. Enzymes are proteins, often described as the molecular workhorses of living cells. They are responsible for all biosynthetic and catabolic processes, as well as being involved in all the dynamics processes, such as DNA replication. They are catalysts, yet are capable of accelerating chemical reactions by factors far in excess of inorganic catalysts, by factors in excess of 1020. Amongst the reactions catalyzed by enzymes, some of the most important are hydrogen transfer, H-transfer reactions, including proton, H-atom, and hydride transfers. The importance of nuclear quantum tunneling in these reactions has been hotly debated for several decades, 14 to 30. But it is well established that H-transfer reactions, whether enzymatic or uncatalyzed, involve some degree of H-tunneling due to the small mass and inherent positional uncertainty of the proton. In this section, we are concerned with the contribution of tunneling to H-transfer during enzyme catalysis, but we should. Quantum Rep. 2021, 3. Also mention the importance of quantum tunneling in biological H-transfer during the bioenergetic conversion of sunlight into proton potentials across cell and mitochondrial membranes. 31.32. For example, a full quantum statistical mechanics approach, 31, has been used to explain a highly efficient proton translocation against the transmembrane voltage, 32, and an observed quantum yield of 2 protons translocated per electron, when a classical description limits the quantum yield to 1, 33. The degree of tunneling during an H-transfer reaction is very sensitive to the barrier through which tunneling occurs and therefore depends on the specific reaction. There is no direct experimental measure for the degree of quantum tunneling during a reaction, but the magnitude of the primary kinetic isotope effect, Ki, is likely to be a reasonable first approximation, 34 to 36. The Ki is the ratio of the rate constant for transfer of a lighter isotope to that for a heavier isotope, for example. Ki equal to Kh, Kd for hydrogen and deuterium, and due to the difference in mass between isotopes of hydrogen, these can have significantly different tunneling probabilities. Chemical reactions are generally described within the framework of generalized transition state theory, TST equation 1, where the rate at a given temperature T depends exponentially on the barrier height, G. The free energy of the transition state relative to the ground state, and linearly on the pre-exponential frequency term, A, which encompasses terms such as recrossing and the transmission coefficient, 203,738. G, KTST equal to AXP, RT, where R is the Boltzmann constant. Enzyme catalysis is primarily driven by a decrease in the barrier height G, to the uncatalyzed reaction, which in turn is thought to mainly arise from electrostatic effects, 39.4. Within semiclassical TST, G is isotopy-dependent, due in part to differences in zero-point energy for different isotopologues. For reactions involving tunneling, the pre-exponential A is multiplied by a tunneling correction factor, KT. This term describes the rate enhancement due to quantum mechanical tunneling and is typically 1 at very low temperatures, where thermal activation becomes insignificant. The rate is determined by a X kappa. T, since both K, T, and G are isotope sensitive, it is not clear whether the experimentally measured key can directly inform on the degree of tunneling. For a purely semiclassical reaction with no tunneling, the key arises only from the zero-point energy differences, which imposes a theoretical upper limit on the key of approximately 7 at room temperature, 41. For some enzymes, where the key is much larger than this limit, for example, SLO1 81, 42, AG 55, 23, and MAG 17, 43, H tunneling is clearly a predominant reaction pathway. But what can be said about the majority of H-transfer enzymes whose key lies below this limit? A range of computational approaches have been developed to model reactions involving nuclear quantum tunneling, such as ensemble-averaged variational transition state theory with multidimensional tunneling corrections, EA-VTSD-MT, 16.44, the quantum classical path, QCP, method, 45.46, and the molecular dynamics with quantum transitions, MDQT, method chapter 17. A very popular method and has been applied to many enzymatic H transfers with Ks ranging from approximately 247 to 51 to approximately 80, 50. For those examples where both the rate enhancement due to tunneling and quantized vibrations, the zero-point energy, have been calculated, there is a strong correlation between these two parameters. 36. This is not surprising, as these effects both originate from the quantum nature of the transferring particle, so that an increase in the quantum mechanical contribution to the reaction will increase both tunneling and the zero-point energy contribution. Given that the K informs on the degree of tunneling, comparing the key of an enzymatic H-tunneling reaction to that of the same uncatalyzed reaction should inform on the contribution of tunneling to catalysis. However, it is usually very difficult to find a suitable reference reaction, and there are only a limited number of examples where experimental data are available for both reactions, 52 to 54. In these examples, the key, Quantum Rep, 2021, 3, 4, of the catalyzed and uncatalyzed H transfer are similar, suggesting a similar degree of tunneling. This is also the conclusion from most computational comparisons, where it is relatively trivial to model the reference reaction in water, 54 to 56, which therefore suggests that tunneling is not a significant factor in the catalytic effect. On the other hand, it is well established that the degree of tunneling, and hence the key, increases significantly with barrier height, as the likelihood of over-the-barrier transfer decreases, so one would expect a significantly lower key in the enzyme-catalyzed reaction. Similarly, the dihydrofolate reductase, DHFR, homologs from mesophilic, each DHFR, 57, thermophilic, BSDHFR, 58, THHR, have very similar Ks, despite the wide range in activation energies. Therefore, it is entirely possible that evolution may have maintained the degree of tunneling even as the barrier height changes. This hypothesis could be addressed within the growing field of paleoenzymology, 59 to 62, by comparing the tunneling contribution in modern enzymes with those from less evolved or extinct organisms, using, for example, ancestral reconstruction. 2.2. Temperature-dependent Ks and the role of fast dynamics. The definitive experimental hallmark of tunneling is a temperature-independent rate constant at low, cryogenic, temperature, where the probability of classical over-the-barrier transfers, the exponential part of equation 1, is negligible. For example, such experiments led to the first evidence of electron tunneling in proteins, 63. However, apart from the practical difficulties in carrying out experiments on enzymes at cryogenic temperatures where processes such as substrate binding will be inhibited, it is more relevant to study tunneling contributions to biological reactions at physiological temperatures. The temperature dependence of enzyme reactions is used to extract relevant kinetic parameters such as the activation enthalpy, H, and entropy, S. And for H tunneling reactions is the temperature dependence of the key, typically measured as the difference in activation energy, EA, or activation enthalpy, H, between isotopologues, Figure 1. Experiments have revealed a wide range of temperature dependencies on the key, from temperature-independent keys, H, equal to 0, to strongly temperature-dependent keys, H the SO. Even for enzymes within the semiclassical limit, the range in H is difficult to account for using semiclassical TSD. However, since the tunneling probability, and hence the key, depends strongly on the tunneling distance, this can be accounted for through differences in donor-acceptor distance sampling at the tunneling-ready configuration, TRC. 64 to 71. The TRC is the molecular configuration arising once an electrostatic environment suitable for tunneling has been achieved through classical reorganization, at which point the tunneling probability can be enhanced by compression along vibrational modes coupled to the donor-acceptor axis on the much faster timescale of barrier crossing, approximately PS. Since the degree of donor-acceptor compression from these modes increases with temperature, this leads to increased tunneling probability and a decrease in key I with temperature as in Figure 1. This donor-acceptor fluctuation is often represented as a promoting vibration, a single harmonic mode which represents the overall donor-acceptor fluctuations along the H coordinate. In this way, the temperature dependence of the chi can be modeled using numerical so-called vibronic models, 72 to 75. Which directly relate the tunneling probability to the tunneling distance. These have been used to model the effect of structural changes between enzyme variants, 737,677, and the role of promoting vibrations calculated from MD simulations, 78 to 80. Additional insight into the role of dynamics in enzymatic H tunneling has come from experiments on the effect of hydrostatic pressure, 80 to 85. Since the zero-point energy, which arises from the high-energy stretching frequency of the breaking bond, is largely invariant with pressure over the experimental range, 86.87, while donor-acceptor fluctuations can be affected. Pressure-dependent chis are indicative of quantum tunneling, 88.89. Unfortunately, no simple trends have emerged between the pressure and temperature dependence of keys, 90. For example, the key of morphinone reductase Mr. was found to increase with pressure while H remained constant while both the key and H. Quantum Rep. 2021, 3. Decreased with pressure in aromatic amine dehydrogenase ARD. These apparently inconsistent sets of data were successfully modeled using atomistic insight from structural data and molecular dynamic simulations. It was found that pressure affects both enzymes differently. In Mr., the promoting vibration becomes stiffer, higher frequency, as the donor and acceptor groups are pushed together, 808,283, while the promoting vibration in ARD becomes softer, lower frequency, despite no significant change in the structure of the active site, 81. Pressure effects are therefore not easily predictable, as pressure acts anisotropically on enzyme structure, 91. Nevertheless, these experiments offer further evidence on the relationship between tunneling and donor-acceptor compressive modes. 1. T Figure 1. The temperature dependence of the key, AA8 or AE, corresponds to the difference in activation enthalpy, R, or activation energy, ANE, between two isotopologues, which are extracted from an Eyring plot, in KT versus 1T, or an Arrhenius plot, in K versus 1T, while the effect on the key and its temperature dependence is now well established. More contentious is the role of rapid donor-acceptor vibrations from an evolutionary perspective and their contribution to the catalytic effect. It has been argued that catalytically important vibrational modes are the same in both the uncatalyzed reaction in solution and the enzyme-catalyzed reaction, 25, and that evolution favors highly pre-organized active sites where only a small degree of donor-acceptor compression will be required to achieve tunneling, 92 to 94. However, since some degree of compression is required for H transfer, even in highly pre-organized active sites, this is true for classical reactions as well as tunneling. It seems likely that evolution has acted not to maximize the contribution of dynamics in H tunneling, but to optimize the efficiency of such modes. This might be achieved by utilizing vibrational modes of active site residues, 95, by restraining the substrate in a conformation which favors specific modes, 96, or simply binding the substrates such that inherent normal modes coincide with the H coordinate, 80. 2.3. A paradigm model system light-activated enzymatic H transfer chemistry. The majority of enzyme reactions are thermally activated, and studies of these systems are limited by diffusion-associated processes, for example, binding of substrates and cofactors, conformational changes in the protein, or product release. In such cases, it is necessary to use rapid mixing approaches to study the reaction chemistry, which often makes it impossible to directly monitor the true rates of any chemical steps during catalysis. Consequently, the reaction chemistry can only easily be investigated over a small range of temperatures and over relatively long milliseconds to seconds timescales. In contrast, if the enzyme is light, activated this problem is removed, as it is possible to trigger catalysis by using a single pulse of light. Light activation thereby allows catalysis to be studied over a wide range of temperatures and timescales, femtoseconds, seconds, 97.98. Hence, light-activated enzyme systems provide a unique opportunity to study tunneling in H-transfer reactions and to investigate the role of fast motions coupled to the reaction. Quantum Rep. 2021. 3. Chemistry. The light-driven chlorophyll biosynthetic enzyme protochlorophyllide oxidoreductase, POR, is one such example and has become an important model system for studying the mechanisms of H transfer reactions, 97.98. The POR enzyme catalyzes the transaddition of hydrogen, 2 protons and 2 electrons, across the C17-4C18 double bond of the chlorophyll precursor, protochlorophyllide, Clyde. The reaction has been studied in detail by using a combination of time-resolved femtosecond seconds spectroscopy, 99 to 106, and cryogenic techniques to trap catalytic intermediates, 97 to 99 billion, 105 million, 107,108, and involves 2 sequential enzymatic H transfer reactions on the microsecond timescale, figure 2. Upon illumination of the Clyde substrate, a hydride anion, 2 electrons and a proton, is transferred from NADPH to the Clyde molecule to generate a negatively charged intermediate, 100,107. This in turn facilitates a proton transfer from either an active site residue or solvent to the C18 position, 99,100. Therefore, POR provides the additional advantage of being able to study 2 separate H-tunneling reactions in a single enzyme system, figure 2. Earlier, laser photoexcitation studies, which investigated the temperature dependence of the keys, showed that both H-transfer reactions proceed by tunneling and are coupled to promoting vibrations in the enzyme-substrate complex, 100. In addition, the keys associated with both the hydride and proton transfer reactions decrease significantly at higher pressures, which suggests that the generic pressure response of hydride and proton transfers are likely to be similar. 90. Figure 2. The light-driven reduction of the C17-C18 double bond of protochlorophyllide, Clyde, catalyzed by protochlorophyllide oxidoreductase, POR. Catalysis involves excited state interactions between the Clyde and protein, leading to sequential hydride transfer from NADPH to the C17 position and proton transfer to the C18 position, with both steps occurring by quantum tunneling. It has been proposed that the role of light in the overall reaction is to overcome the barrier for the highly unfavorable hydride transfer chemistry, 100. Upon illumination, an electron-deficient double bond is generated in the plied excited state, which promotes the nucleophilic attack of the negatively charged hydride anion from end AD 103. This hydride transfer reaction in POR has recently been shown to occur in a stepwise manner and involves an initial electron transfer from NADPH to the excited state of Clyde, followed by a proton-coupled electron, or H atom, transfer to form a deprotonated product, anion, 104,105. This is the first report of a stepwise hydride transfer in enzyme catalysis, rather than as a single chemical entity, which has only been possible to observe due to the very fast time resolution that can be obtained from triggering the pore reaction with a short laser pulse. 109. The rate and temperature dependence of the key of the hydride transfer chemistry is conserved across all light-dependent pores from plants, algae and cyanobacteria, 100 and 2106. In all pore enzymes studied to date, there is a breakpoint at minus 27 degrees Celsius in the temperature dependence of the key for hydride transfer, 100,106. Above this temperature, the key I is temperature dependent, which implies that the hydride tunneling reaction is reliant on promoting vibrations. In contrast, below minus 27 degrees Celsius, the key becomes independent of temperature, suggesting that these promoting vibrations are quenched. In addition, the rate of hydride transfer is independent of the bulk solvent viscosity, and it is therefore likely that Quantum Rep. 2021 3. 7. The hydride transfer reaction is controlled by localized dynamics in the active site, rather than any large-scale protein motions, 101,106. The active site architecture is proposed to be conserved in all PORs, and minor changes to the active site geometry are known to compromise hydride transfer by substantially affecting the efficiency of the photochemistry, 99,103 to 105. The stringent structural requirements and optimal positioning of NADPH and Clyde in the active site are thought to be essential to facilitate this difficult light-driven hydride transfer chemistry in all PORs, 100,106. This has been confirmed by a recent structural model of the pore-plied NADPH ternary complex, which suggested a donor-acceptor distance from NADPH to the C17 plied of approximately 4,5A, although this may well change upon excitation of the Clyde molecule, 110. Following hydride transfer, there is a proton transfer reaction to the negatively charged intermediate plied from either a conserved TIA residue or surrounding solvent to generate the final product, 99,100. In contrast to hydride transfer, there are major differences in the dynamic control of the proton transfer reaction between pore enzymes from different species, 102,106,108. In cyanobacterial pores, proton transfer is reliant on solvent dynamics, and the associated key is temperature dependent, consistent with there being a complex network of extended protein motions that are coupled to the proton tunneling reaction, 101,102,106. Conversely, proton transfer in plant pores is much faster and is not dependent on a solvent-coupled dynamic network, 102,106,108. This suggests that the proton donor is located in an optimal position for proton transfer in plant pores, but a degree of protein reorganization is likely to be necessary to optimize the proton donor-acceptor distance in the cyanobacterial enzymes, 102,106,108. A recent structural model of the cyanobacterial enzyme appears to confirm this hypothesis, as a donor-acceptor distance of approximately 5A was observed between the putative conserved tyrosine proton donor and the acceptor position of Clyde, 110. It is also likely that a large proton relay pathway is coupled to this tyrosine residue, which consists of a water network coordinated by a number of active site residues, 110,111. Overall, the work on POR has highlighted the advantages of studying H-tunneling reactions in a light-activated system and has shed light on the mechanism of H-transfer reactions and the role of fast protein dynamics that are coupled to the reaction chemistry. 2.4. Conclusions. To date, a wealth of information has been gained on the role of tunneling and fast dynamics in enzymatic H-transfer reactions from studies of the temperature dependence of the observed rate and key. The importance of tunneling from an evolutionary perspective and the role of dynamics in promoting H-transfer remains contentious, and the temperature dependence of keys does not, by itself, indicate the extent to which the protein or solvent are involved. Hence, alternative methods to probe tunneling reactions and the role of fast protein dynamics are being sought. Here we have described how POR is a paradigm model of a light-activated enzyme system, which may facilitate advances in the studies of the relative contribution of nuclear quantum effects in H-transfer chemistry. By comparing experimental observations and modeling data for POR analogues from different species, and taking it further to look at enzymes from more primitive or extinct species using paleoenzymology, this system might shed light on the evolution of enzymatic H-tunneling. 3. Quantum effects in photosynthesis. Photosynthetic organisms harvest light using networks of chromophores that absorb the light and efficiently funnel solar energy toward a reaction center where charge separation occurs. Nature's complex system of chromophores form an antenna to harvest light, separating the light harvesting from the charge separation, a design principle that researchers would like to copy in next-generation technologies. Photosynthetic growth occurs via the conversion of molecular excitations into long-lived chemical energy in the form of adenosine triphosphate, ATP, and reduced nicotinamide adenine. Quantum rep. 2021, 3,8-dinucleotide phosphate. These energy-rich molecules fuel the fixation of CO2 via the Calvin-Benson-Basham cycle, 112. The overall process of photosynthesis is fairly thermodynamically inefficient, with a practical maximal efficiency of light to biomass conversion of below 10%, 113,114. However, the initial step of capturing a photon with a light-harvesting molecule and the transportation to a reaction center happens with a quantum efficiency of near unity. At the same time, these processes are remarkably robust, 115. This highly evolved process has been proposed to take advantage of quantum mechanical effects, despite the warm and noisy environment in which photosynthesis operates, 116. Understanding the mechanisms operative within the light harvesting antenna and subsequent transport to the reaction center within the photosynthetic organism may help to define the next generation of green energy solutions and optoelectronics. 3.1. Theory and experimental studies. Quantum dynamics has been proposed to play an important role in the light reactions of photosynthesis, and these ideas, though contentious, have received support from both theoretical and experimental studies. Photosynthesis light harvesting complexes generally consist of extended arrays of pigments fixed to a rigid protein scaffold. The dense packing of chromophores leads to a range of interactions and coupling strengths within each complex. After absorption of photons by a pigment or group of pigments contained within a given light harvesting complex, LHC, the transfer of energy occurs among these molecules. For chromophores with large separations and a low Coulombic interaction, Forster resonance energy transfer, FRET, theory, 117, provides an accurate description of the energy transfer as a series of incoherent hops. For strongly coupled chromophores with relatively weak coupling to their surroundings, Redfield theory provides a good description of energy transfer as relaxation within the coupled chromophores. More complex descriptions are needed when multiple types of couplings are relatively strong or where specific motions within the bath are important. Regardless of mechanism, energy is ultimately trapped into a reaction center, RC, where charge separation takes place. In a bacterial system, electronic transitions are guided by a downhill energy funnel from higher energy states to lower energy states, but in higher plants, such energy separation is not obvious. The description of energy transfer in photosynthesis has a very long history, from coherent descriptions in 1932 by Perrin, 118, to Forster's seminal work in 1945, 119. However, it has become clear that Fret alone cannot be employed to explain the process occurring in photosynthesis due to a significant coupling strength outside of the Fret regime between their photoexcited chromophores, 120 to 125. Moreover, random energy transfer between densely packed antennae pigments would lead to high losses to the environment before reaching the RC, resulting in a lower photosynthetic quantum yield than observed, 126 to 128. Highly packed pigments, for example, in the light harvesting complex 2 of purple bacteria, LH2, demonstrate stronger couplings and interaction strengths occurring between molecules wherein the excitation is not isolated to a specific pigment, but rather delocalized in the form of a Frenkel exciton, 129,130. Given the wide range of structures seen in the natural world, it is not surprising that the Redfield and Forster models that describe strongly coupled and weakly coupled regimes respectively are inadequate to describe the coupling of all chromophores within photosynthetic complexes. 121. Many systems appear to occupy an intermediate regime that is not captured well by theories describing either extreme of coupling strength. 131. Modified versions of these models have been built to account for several important phenomena seen during experimental observations. 132 to 134. For specific mathematical models of the bath surrounding the chromophores, the problem has been solved exactly by Ishizaki and Tanimura, using hierarchical equations of motion. 135,136. A more general pseudomode method has been introduced by Lambert and CO workers that includes Matsubara terms and is readily suited for low-temperature studies. 137. Other exact methods for arbitrary harmonic bath models, Quantum Rep 2021-39, have been introduced by Plenio and CO workers, exploiting density matrix renormalization group methods. 138 to 141. With the advance in new spectroscopic techniques such as two-dimensional electronic spectroscopy, 2DES, phase-sensitive investigations into the nature of the energy transfer taking place in LHCs could be performed. An accessible introduction to the theory and data analysis of 2DES signals are provided by Gelzinis et al. And Wang et al. 142,143. In short, contrasting with the classical pump probe spectroscopy, in 2DES the excitation frequency is resolved, permitting more accurate maps of energy transfer in spectral congested systems. After the excitation of the absorption electronic bands of a given system, recorded on the excitation axis, omega-excite, the system evolves during a waiting time, T2 or T-lay, having its current state recorded on a detection axis, omega-detect, to generate a correlation map. On this map, diagonal peaks correspond roughly to the linear spectrum of the system, and the cross peaks to the interactions occurring between their absorption bands, arising early, transitions occurring between common electronic orbitals, or later, energy transfer, and possibly oscillating with the time. Figure 3a. In this way, this technique has been successfully employed during the last decades for obtaining information on energy transfer pathways, interactions occurring between the system and the surrounding environment. Bath, 144,145. Energy levels found within a photosynthetic complex are thought to be tuned to the environmental bath composed of protein and solvent interactions. 146. However, there is some debate over where the line between system and bath is drawn. 147. As progress in the field continues, we see how the system now encompasses more of what was typically previously seen as the bath dynamics. The tuning of spectral properties of the pigments due to their coupling, as well as with the protein and solvent environment, facilitates ultra-fast energy transfer, interactions between chromophores, and many other aspects related to the temporal dynamics of LHCs. The application of 2D is, at cryogenic temperature, 77K, to track energy transport within the Fenner-Matthews-Olson FMO complexes of the green sulfur bacterium Chlorobium tepidum by Engel et al. In 2007, was one of the key advances of modern quantum biology, as it provided evidence for the possible involvement of quantum dynamics occurring within this system. 148. In this work, oscillating crosspeaks arising at early waiting times up to 660 Fs were attributed to electronic coherences occurring between the chromophores enclosed within the protein. These ultra-fast quantum beats witness the rapidly changing populations of excited states within the system. The occurrence of strong coupling between Frenkel excitons formed after the light absorption and the consequent delocalization of their excitation is by itself a quantum effect capable of modifying energy transfer pathways occurring within light harvesting complexes due to modifications of their collective dipole configurations. However, it is possible that quantum mechanics are playing an even bigger role in the process, by allowing the system to sample different pathways simultaneously through the superposition of states and finding the most efficient path to the reaction center, constituting what researchers have been calling a quantum walk. Quantum entanglement has also been proposed to be playing a role in the system, 149, although this is more conjectural. The landmark paper by Engel et al., 148, received many critics from specialists in the field, which argued that the oscillations observed in the work should be attributed vibrational modes on the electronic ground state of the protein system, 150. Nevertheless, it also provoked a flurry of similar studies investigating the concept of long-lived coherence in photosynthesis. Most studies have focused on the light-harvesting complexes of purple non-sulfur, LH1RC and LH2, and green sulfur, FMO, bacteria. For example, in 2007, Lee et al. Used two-color electronic coherence photon echo spectroscopy, 2-SECP, to characterize coherences between chromophores present within the isolated reaction centers, RCs, of Rhodobacter spheroids, 151, and similarly detected quantum beats persisting for longer than 400 Fs, at 77K. The study also provided evidence of strongly correlated and long-lived nuclear modes, suggesting that the energy levels of the chromophores were being modulated by vibrational modes. The finding that the vibrational modes were of similar, free, quantum rep, 2021 term 3, 10, frequencies to those of the excitonic energy gap suggested a vibronic scenario in which nuclear vibrations were fine-tuned to enhance electronic energy transfer, 152,153. A theoretical study of the FMO complex, 154,155, came to similar conclusions. Vibronic transport has also been shown to increase the energy transfer by 10% in the LHC of the algae Chromonas mesostigmatica, 156. The possibility of resonance between electronic and vibrational states leads to some interesting and non-trivial dynamics of mixed character that are thought to influence energy transfer in both the antennae complexes and reaction centers. 154,157,158. For additional studies of other model LHCs, we refer the reader to Refs 159 to 161. Figure 3. A. Time evolution of signal amplitudes obtained with 2 DES. A cross peak is shown oscillating in the frequency equivalent to the energy difference between the two states in the diagonal. A Fourier transformation is applied to extract the frequency of the beating signal. Adapted from Ref. 130. B. 2D spectra obtained for the FMO complex at different temperatures at T delay equal to 400 Fs. The oscillations obtained for a given spectral position, white circle, possess a similar phase and frequency in all cases with shorter lifetimes at physiological temperatures, 277 K. Adapted from Ref. 162. A key question is how relevant long-lived coherences measured in the laboratory are to photosynthesis taking place within noisy biological environments. 150,163,164. However, A key concept that has emerged from several studies is the role that the environment may play in actually enhancing, rather than destroying, quantum coherence. Experiments carried on the FMO complex at physiologic temperatures, 277 K, demonstrated quantum beats that persisted for 300 Fs, figure 3b. A similar study of LH2 of Rhodopseudomonas acidophila, 10,050, demonstrated quantum beats persisting for at least 400 Fs, long enough to play a non-trivial role in the energy transfer. 162,165. Another room temperature study of the LH2 of Rhodobacter spheroids provided evidence of a role for environment-assisted quantum interference between multiple energy transfer and relaxation pathways in a phenomenon often called quantum phonon antennae. 166,167. The findings were consistent with theoretical studies that predicted the enhancement of energy transport at intermediate noise levels, corresponding to those characteristics of room temperature, when the dephasing frequency is tuned to the excitonic energy gaps and couplings. 168 to 170. Although it has been argued that results, such as these, provide evidence of classical, rather than quantum behavior, from his own work on the LH2 of Rhodoblastus acidophilus, formerly Rhodopseudomonas acidophila 7050, Zhang argued that quantum mechanical dynamics cannot be neglected when modeling transfer dynamics between LHCs is separated by more than 1 nanometer. Nevertheless, the interpretation of these spectra remains controversial, with several researchers opting for an interpretation of a mixture of excitonic dynamics and fret involved in the energy transfer. 171. While extensive quantum biology studies focus on excitation energy transfer, biology also exploits proton transfer, and there are suggestions that the quantum efficiency of this mechanism may also be enhanced by quantum effects. 172. Halobacteria possess quantum rep. 2021, 3, 11. Retinal-containing protein complexes called bacteria hodopsins that function by pumping protons across their membrane upon absorption of a photon. 173,174. The light-driven proton transfer mediated by these proteins generates a proton gradient, which drives the synthesis of ATP by ATPases localized in the membrane of these bacteria, constituting an important alternative source of energy. 174. These complexes are currently being commercialized in the form of membrane patches obtained from Halobacterium salinarum and are employed during the production of artificial retina, photosensitive and erasable material for optical information, recording and processing, and electronic ink for laptop display. 175 to 178. A simple model of light-induced proton pumps in artificial photosynthetic systems was studied by Ghosh et al., in which a photon-to-proton quantum yield of the model system was analyzed as approximately 55%, 179. 3.2. Bio-inspired synthetic light harvesting systems. 2D's experimental spectra of photosynthetic complexes can easily become congested and thereby difficult to interpret. To counteract this, investigations into energy transfer dynamics in photosynthetic complexes have recently been conducted in simpler bio-inspired synthetic systems constructed with biological and synthetic parts. Such hybrid biotic-abiotic systems are also of interest because they could provide the prototypes of next-generation green energy technologies, including incorporation into solar cells. 180,181. Alongside theoretical modeling of the quantum dynamics of both charge and energy transfer in an artificial system. 182. Such an approach can complement lab-based investigations. 183. Design principles of lab-based bio-inspired systems typically utilize a biologically relevant chromophore system anchored to a scaffold through various covalent and non-covalent functionalizations that are designed to mimic chromophore pigment interactions. As described above, coupling of chromophores to their environment in a LHC is thought to play a vital role in facilitating fast energy transfer. In one study of energy transfer from carotenoid peridinin donor to acceptor chlorophylls from marine dinoflagellates, 184, the energy gap between donor and acceptor was tuned to modify, and thereby investigate, the role of electronic and vibrational coupling. The data-rich technique of 2Ds demonstrated that minor changes to the chlorophyll functional groups, for example, the formal group, led to faster decoherence and lowered energy transfer efficiency within the complex, 184. An easily modifiable scaffold, such as the tobacco mosaic virus capsid protein, offers a highly versatile base from which chromophores can be attached at precise locations using linkers of varying length and rigidities. In one study, rotationally rigid linkers were shown to have a large influence on the lifetime of exciton correlations, 185. To further investigate linker chemistry, the distinct vibrational properties of chemically functionalized carbon nanotubes was utilized to probe the effects of vibronic coupling on coherence and energy transport within various chromophore heterodimers, 186. In addition, room temperature quantum beatings associated with strong vibronic coupling was demonstrated in a system of multichromophoric synthetic heterodimers with rigid linker chemistry, 187. Dimerized chromophores provide another means to investigate variables affecting interchromophore coupling and long-range efficient charge transfer, as well as representing the reaction center photo-induced charge separation of the special pair. Using both symmetric and antisymmetric dyads, the extent of excitonic coupling on coherence times can be investigated without the typical experimental challenges associated with direct in vivo experiments on reaction centers and their many reaction pathways. 186. Synthetic model system of bacterial photosynthesis. Absorption spectra of bacteriochlorin BC, dimers as seen in Figure 4a, were measured with phenylene bridges of varying lengths, 188. Stronger dimer interaction due to the shorter phenylene bridges led to excitonic splitting of bands and faster relaxation, as seen in the time-resolved photoluminescent spectra in Figure 4b. Additionally, the excited state dynamics of the stronger coupled system was drastically modified by bridge length and solvent choice. Efficiency of Quantum Rep. 2021, 3. 12. Charge transfer decreased with increased bridge length by nearly 1 order of magnitude with each phenylene addition, 188. Dynamics of the system was investigated by 2DEs and was shown to be consistent with coherence, having both electronic and vibrational components, 189. B. Figure 4. Inserting phenylene bridges between two chromophores shown in A has a profound effect on the coupling and excited state dynamics of a dimer system. Faster relaxation is seen in the time-resolved photoluminescence spectra, B, when the coupling is stronger and the pigment spacing is smaller. This study reflects properties seen within special pairs in cyanobacteria. Adapted from Ref. 188. Considerable efforts have also been invested in projects aimed at building more complex and bio-inspired wholly synthetic systems, although larger aggregates are still some way off the complexity of a biological light harvesting complex. Drawing inspiration from biological light harvesting complexes, chlorosomal J aggregates have been employed as a mimic of the chlorosomal light harvesting units within green bacterial photosynthesis and exhibited efficient singlet energy transfer using a biologically relevant solvent. 190,191. Elsewhere, the rigid scaffold chromophore interactions of natural light harvesting systems have been mimicked with cyanine dye-derived host solid state matrix. Focusing on the structural similarities to chromosomal assemblies in green sulfur bacteria, capable of low-light photosynthesis, the solid-state environment was found to modulate vibronic coupling of the system and direct energy transfer. 192. Agglomeration of dyes into nanotubes and structured bundles bio. Inspired green bacteria facilitated longer exciton diffusion lengths and wider exciton delocalization consistent with theory. 193 to 195. Using similar systems, Vibronic coupling was shown to be the origin of long-lived beating signals in tubular and nanoring artificial light harvesting architectures that mimicked interpigment distances, 196,197. 3.3. Conclusions. The detection of quantum effects in photosystems persisting for long enough to be considered non-trivial has led to substantial progress over the last decades. The subject of coherence in photosynthesis is an ongoing contentious issue, with comprehensive previous reviews centered on both experimental and theoretical advancements, 12,130,198 to 201. Although there is no doubt that coherence is present in LHCs over short time scales, proof of a functional role for quantum coherence in photosynthesis still eludes researchers. Recent research has instead highlighted an interplay between both quantum and classical, Quantum Rep. 2021, 3.13. Mechanics in photosynthetic systems. Whilst controversies continue concerning the origin of photosynthetic coherence, measured by 2DS, future advances in spectroscopy and other observation techniques provides the best hope of resolving differences in interpretation. 165,200,202. Practical experimental approaches to verify the quantumness of the observations are now being developed and implemented. 203 to 205. Moreover, use of wholly or partly synthetic systems can provide greater simplicity coupled with finer control of the structure of the system, as well providing a simpler theoretical model. It is hoped that further development of these tools will finally elucidate the role that quantum mechanics plays in the most important chemical reaction on our planet. 4. Magnetic field effects on spin-dependent reactions in biology, candidates and constraints. Radicals and other molecules with unpaired electron spins, that is paramagnets, are found across biology, 206, and can be highly reactive. At high concentrations, free radicals such as reactive oxygen species, ROS, can overwhelm cellular coping mechanisms, resulting in oxidative damage and even cell death, 207,208. By contrast, many biological systems have evolved to harness the power of this reactivity. Free radicals can play important roles in the immune response, 209, signaling, 210, and mitochondrial respiration, 211. Transient radical intermediates are also common in enzyme-catalyzed reactions, 212, and photoreceptors, 213. If the unpaired electron spins of interacting paramagnetic species become correlated, and if their chemistry is in some way dependent on the resulting spin state, their reactions can be influenced by the presence of magnetic fields, MF. 214. The most prominent example of this is the radical pair, RP, model of animal magnetoreception, 215, which is also among the most high-profile putative examples of quantum biology. Not only are paramagnetic species widespread in biology, but sources of MFS are also commonplace in our environment. They range from very weak MFs like that of the Earth, approximately 50 UT, to much stronger fields generated by MRI magnets, 1 trillion. One might therefore assume that MFEs on spin-dependent biochemical reactions are likely to be similarly common. There are several conditions that must be met, however, for such MFEs to manifest, for a checklist see Figure 6 AD. In this review, we will discuss the relevant constraints in the context of candidate systems for biological MF sensitivity, from the putative magnetoreceptor, cryptochrome, 215, to lipid peroxidation, 216,217. Our intention is neither to give a comprehensive account of all biological MF and spin effects observed to date, nor to simply provide another overview of the RP hypothesis of animal magnetoreception. Comprehensive reviews of these topics are available elsewhere. For example, 215,218 to 224. Instead, we will use a selection of examples to scrutinize these constraints and thus assess how widespread MFEs are likely to be in biology. First, we will briefly describe how a RP reaction can be magnetically sensitive, See also Box 1 and Figures 5 and 6AB. For more detailed accounts, we direct you towards some excellent reviews, for example, 214,225,226. When a pair of radicals is generated simultaneously from non-radical precursors, for example, through an electron transfer reaction, the unpaired electron spins are initially correlated, or even entangled, with a multiplicity determined by that of the precursor. The paired spins are then either created in singlet, S, anti-parallel spins, or triplet, T, parallel spins, spin states, figure 5, which have a different reactivity. Often only the SRP can recombine, figure 6A, or if low-lying triplet product states exist, S and T recombination gives rise to different products. For MFA to manifest, the initial spin state must be converted to states with different chemical reactivity, for example, S to T or vice versa, in a MF-dependent fashion. In many cases of interest, this interconversion between spin states of the RP is coherently driven by local magnetic interactions of nuclear spins within the radicals, known as the hyperfine couplings, HFC, figure 6b. Because this process is magnetic in origin, the relative populations of S and T RPs, and hence the outcomes of RP reactions, can be perturbed by external MFS. Spin relaxation processes can abolish spin correlation and thus remove the magnetic sensitivity of the system. Further MFEs can result, however, if the MF impacts on the relaxation dynamics in competition with Quantum Rep. 2021, 3.14. Spin-selective recombination processes, known as the relaxation mechanism. If the spin correlation is preserved for times longer than about 1 US, the reaction can in principle be sensitive to extremely weak MFS comparable to the geomagnetic field, approximately 50 OT. That this limit is practically attainable has been demonstrated by the magnetosensitivity of RP reactions in purposefully engineered RP systems, such as a carotenoid porphyrin fullerene triad, 227. Box 1. Some key concepts. Spin correlation. Spin correlation means that local measurements for each spin are no longer independent, often pictorially represented as either parallel or antiparallel spins, figure 5, and can refer to both classical and quantum correlation. Purely classical RMP spin states include the T plus triplet state, for which the z direction of spin precession for both electrons are pointing up. An example of a maximally entangled state is the S spin state of the RP, where states of opposite electron spin polarization at the two radicals are in a coherent superposition. Entanglement. A state of two or more particles is entangled if it is in a coherent superposition state, that is, it cannot be expressed as product state of states pertaining to the individual particles. Consequently, quantum states of multiple subsystems of an entangled system cannot be described independently of each other, even though the subsystems are spatially separated. Entanglement is a uniquely quantum mechanical resource in effects such as quantum teleportation or fast quantum algorithms. Unlike spin correlation, it is not critical to the manifestation of MFEs. Spin multiplicity, the spin multiplicity of the unpaired electrons of an RP, is the number of quantum mechanically allowed orientations of the spin vectors and reflects the total spin angular momentum. Coherence. The time evolution of the RP spin state is said to be coherent when the oscillatory interconversion between spin states has a regular frequency and is driven by stable local MFS. Spin state interconversion can also be impacted by incoherent relaxation processes, driven by random magnetic fluctuations, which abolish spin correlation and take the system into a pair of statistically independent, randomly oriented spins. Quantum coherence involves superposition of Rivy-Dobasis states. 15. Figure 5. 5. Vector representation of off-correlated electron-to-electron spin status inina RPA RP quantize along the axis over an applied MEF MF B0 in. Inia Keach case Thethian paradun. Paired electron-nectron spin vector for foro keek radical oaf the pair is represented bbia black arrow. The theconescon stratila straight the precession of the expectation value of h each spin vector about the direction of B0. S denotes the singlet state and T denotes the three triplet states. Spin vector about the direction of B0. S denotes the singlet state and T denotes the three triplet states. In general, the spin state of the RP is a coherent superposition of these states, whereby the probable in general, the spin state of the RP is a coherent superposition of these states, whereby the probabilities ities of finding the system in a particular state oscillate with time as a result of, for example, hyperfine and Zeeman of finding interactions. The system in a particular state oscillate with time as a result of, for example, hyperfine and Zeeman interactions. Quantum Rep. 2021, 3, 15. A. Figure 6. Cont. Quantum Rep. 2021, 3, 16. Figure 6. Checklist of the constraints for a magnetically sensitive RP reaction. A. Example schemes illustrating the generation of a S-borne, left panel, red arrows or T-borne, right panel, green arrows, geminate RP by electron transfer and the subsequent spin and reaction dynamics. In both cases, excitation, HNU, of the S-ground state of the electron donor chromophore, D, initially populates the S-excited state, wonder, if the electron is transferred to the acceptor molecule, A, from here, the RP is S-borne, wand plus A. The excited S state might instead undergo intersystem crossing, ISC, to a locally excited T state, 3D, before electron transfer. RPS resulting from here are T-borne, 3D plus A. When the radicals of the pair diffuse apart, see panels CD, or are sufficiently separated a priori, they can undergo ST interconversion, S, which is typically driven by differences in HFCs in each radical of the pair, see panel B, the ability of each spin state to recombine, that is, back electron transfer, or form new reaction products, spin-independent, in competitive reaction dynamics, is spin-selective. B. Schematic illustration of HFCs in a SRP formed by electron transfer. Each HFC is between the magnetic moment of surrounding nuclei, N1 and N2, with the magnetic moments associated with the unpaired electron spins in each radical. C. Variation of energy levels, E, of the RP spin states as a function of applied MF, B, Zeeman interaction. The energies are plot, both for a RP with black and without, cyan, a significant exchange interaction, J, R, which varies as a function of the separation, R, between the radicals of the pair. The plots are offset for clarity. D. An illustrative plot of the energy, E, that corresponds to 2JR of example RP spin states as a function of separation, R, between the unpaired electron spins. SRPs, red and blue arrows, TRPS, green arrows, assuming the S configuration is bonding. E. Conceptual diagram illustrating the window of magnetic sensitivity on the timescale for the spin dynamic evolution for a typical organic RP lifetimes that are too short, e.g. due to rapid recombination, do not allow time for significant spin state evolution, and thus do not exhibit magnetic sensitivity. For very long lifetimes that outlast the coherence lifetimes, the electron spins are fully equilibrated and no more susceptible to weak MFS. Even if the RP reaction and spin dynamics do fall within the so-called MF-sensitive region, the reaction evolution pathways must still be spin-selective. That is, at least one reaction product must be formed in a spin-selective process. This will not be the case if the kinetics of spin-independent radical quenching out compete those of the spin-dependent recombination, for example, for B12-dependent eel. 4.1. The question of thermodynamics. For a stimulus to impact any chemical system at equilibrium, it must exceed the thermal energy at the relevant temperature, which equates to kBT, per degree of freedom. This appears to represent a significant barrier to biological MFEs. It is one reason why magnetic resonance imaging, MRI, is such a powerful technique, since static and radiofrequency MFS interact only weakly with biomolecules, compared to, for example, visible light, allowing detailed images to be captured non-invasively from deep inside the body. the effective interaction energies between electron spins, and the kind of weak MFS encountered in our environment, 1 MT, are at least 5 orders of magnitude lower than KBT at physiological temperatures 219,228 to 230. Chemical bonds are stronger still, 10 to 100 x KBT, leaving MFS little chance to impact biochemical reactivity by changing the free energy of the system. Yet even very weak MFS, ranging from static to radio frequencies, can and do influence chemical reactions, 214,231. Even those as weak as the Earth's MF, 227. Part of the reason is likely to be that the KBT limit only strictly applies to systems at thermal equilibrium, and the spin-correlated RP described above is, at least transiently, far from thermodynamic equilibrium. This is often a direct result of the RP generation, where S&T precursors, Quantum Rep, 2021, 3, 17, generate RPS initially in S and T states respectively as described above, in essence reflecting the Pauli exclusion principle, which for close electrons dictates the system's energy on scales hugely exceeding KBT. Due to its non-equilibrium nature, the spin dynamics of RPs are therefore not bound by the KBT argument until the system relaxes to a Boltzmann distribution of spin states, giving the MF a window of opportunity, Figure 6e, to change the population of RPEs in each spin state and hence the reactive fate of the system. If the coherent evolution is faster, or at least comparable to the spin relaxation time, MFEs can thus manifest despite all interaction energies governing the spin dynamics being minuscule relative to KBT. 4.2. Is spin correlation common in biology? Perhaps the most common route to spin correlation is the simultaneous generation of RPs from non-radical precursors known as geminate RPS. Electron transfer reactions between neutral diamagnetic donors, D, and acceptors, A, produce geminate radical ion pairs, D plus A, produce geminate radicals and can be triggered by light. For example, photogenerated RPS in the flavin-dependent photoreceptor cryptochrome, 232, is thought to be the reason why magnetoreception in birds, 233, and Drosophila, 234, is light-dependent. Other examples include light, oxygen, or voltage sensing, LOVE, domains, 235, and the light-driven enzymes protochlorophyllide oxidoreductase, CF, section 2.3 and figure 2, 109, and the cryptochrome-related DNA photolase, 236. Electron transfer to generate geminate RPS can also be triggered thermally, including by enzymes such as cytochrome 450 piezose reductase, 237, and monoamine oxidase, 238. Geminate RPS can also be generated by bond homolysis, as exemplified by the biochemistry of vitamin B12, 239. Upon substrate binding to its dependent enzymes, the CO-C bond in coenzyme B12, 50-deoxyadenosylcobalamin, 8-OCBL, breaks homolytically to produce a spin-correlated COB, 2-alamine-adenosyl RP, 240. The same geminate RP is generated following photolysis of ADO-CBL and its related B12 cofactor, methylcobalamin, MeCBL. The same geminate of both ADO-CBL and MeCBL show magnetically sensitive RP reaction dynamics, 242, that the photolysis of MeCBL also generates spin-correlated methyl-methyl RPs, 243. In contrast to geminate RPS, here the spin-correlated state is generated following the random encounter in solution of freely diffusing, uncorrelated radicals, known as F-pairs. Although initially uncorrelated, spin-selective recombination processes during their initial encounter mean spin correlation can nonetheless emerge. For example, S encounters lead to recombination in one of the random encounters, leaving the T states, resulting from 3 of the encounters, poised to undergo magnetosensi 4 type spin dynamics. F pairs have also been implicated in the magnetic sensitivity of lipid peroxidation, 216,217,244, which recent model calculations have attributed to the recombination of two lipid peroxyl radicals via the Russell mechanism. That is the recombination of two peroxyl radicals via an unstable tetroxide intermediate. For most of the examples discussed above, there is direct evidence for the radical intermediates. In some cases, such intermediates are, instead, inferred, and it is here where magnetic spin effects have the potential to serve as mechanistic probes. One remarkable example is the magnetic isotope 245 to 247 and MF effects 248 reported on enzymatic phosphorylation. Here, the authors invoke the PO3 Mg plus radical ion pair to explain their observations. There is little precedence in chemistry for a Mg plus radical, however, and attempts to reproduce some of the reported effects have been unsuccessful, 249. Elsewhere, radiofrequency MFS has been reported to increase levels of cellular ROS, 250,251. The authors propose FADH, superoxide RP produced by flavoenzymes, and that a 7% MHZ MF is resonant with flavin radical HFCs. Flavins generally provide a rich source of MFEs due to their ability to form RPs in photo-induced electron transfer reactions with various electron donors, 252, both from the S and the T excited states, 253. Recently, the endogenous autofluorescence of HeLa cells has been found to respond to external MFS due to mitochondrial, triplet-borne, flavin-containing RPS, 254. Quantum rep, 221. What drives coherent spin-state evolution, and what coherence lifetimes are necessary? In the simple, semi-classical picture, the coherent S-T interconversion can be attributed to different precession rates of the unpaired electron spins in their effective local MF. At most environmental MF magnitudes, 1 trillion, this interconversion is primarily driven by HFCs, figure 6b, 225. In the vast majority of cases, the members of biological geminate pairs will be different chemical species where the nature and extent of HFCs are different. In this case, coherent ST interconversion results from the differential dynamics of the electron spins in the different local fields. In weak MFS, this process involves all four electronic spin states. Effects of moderate MFS, tens hundreds of MT, on typical RP reactions result when the T1 spin states are removed in energy by the Zeeman interaction, figure 6c. Ultimately, the HFCs can no longer drive interconversion between the T1 and S spin states, which alters the S and T populations relative to zero applied MF and hence the recombination probability. Interestingly, even the recombination of identical radicals can be magnetically sensitive if the nuclear spin configuration of each radical is different at the point of encounter. Most biological radicals are based on organic molecules, which means the magnitude of the HFCs, and by extension the spin-state mixing frequency, fall within a relatively narrow range, extending to tens of MHZ, 214,225. Suggesting that the main effect is expected for MTMFs. There are some notable exceptions, however. Relative to its organic partner, the cobalt-centered radical in the COB, 2-alanine alkyl RPs from B12, has a substantial HFC. This results in sub-nanosecond, NS, spin conversion times compared to tens of NS for typical organic RPS, 255, and the highly unusual observation of MFEs on the ultra-fast RP reaction dynamics following photolysis of EDIO CBl 256. MFEs have also been reported on the ultra-fast decay kinetics of photoexcited bacteriochlorophyll, but this time from exposure to very high MF, 9 trillions, and resulting from the triplet mechanism and PS electron spin relaxation, 257. There are also examples where one of the suggested paramagnetic RMP partners has no HFCs at all. In the putative magnetoreceptor, cryptochrome, the most commonly studied RP comprises the flavin semiquinone and tryptophanyl radicals from the photoreduction of AD, 215,258,259. It has been proposed, however, that the magnetically sensitive Rp is instead the flavin semiquin superoxide radical, which can be formed during the reoxidation of the Fad dihydroquin, 260,261. The superoxide radical is devoid of intrinsic HFCs, which conveys possible benefits in the context of sensing a very weak MF like that of the Earth's, for example, a superior directional sensitivity, 262,263. There are also some potential drawbacks, as freely diffusing superoxide is subject to fast spin relaxation, which will outcompete the coherence time needed for sensitivity to weak MFS to manifest, 264. In the context of magnetoreception, this discrepancy has led to the postulated involvement of an as of yet unidentified radical, Z, instead of superoxide, which is again devoid of HFCs but relaxes comparatively slowly, 262. For sensing the geomagnetic field, spin coherence times must be at least 1 us, so that the Lama precession in the geomagnetic field can significantly impact on the dynamics. This bound appears to be in agreement with predictions of the spin relaxation time of flavin semiquinone tryptophanol RPs in cryptochrome at physiological temperatures, which are expected to occur on the order of 1 to 10 us, 265 to 267. At much higher MF exposures, like those produced by MRI magnets, 1 trillion, spin state mixing can also be driven by the difference between the molecular G values, dimensionless factors that quantify the magnitude of the magnetic moment associated with spin, of the paramagnetic pair members, as discussed in the caption of Figure 6. Effects of MF of up to 18 trillions, owing to the so-called G mechanism, have been observed on reactions involving the NO and superoxide radicals, 268, both of which have important signaling roles in biology. Much like its HFCs, the molecular G value of the B12 COB2 alanine radical is substantially larger than that of its alkyl radical partner. Again, this results in the observation of unique MFEs for B12 photolysis, this time with the first observation of the G effect at MF well below 1 trillion, again on the ultra-fast timescale, 256. For typical organic radicals, however, G is quantum rep 2021, 319. Tiny, and this mechanism is thus negligible in weaker MFS. Huge spin-orbit interactions could, at least in principle, give rise to more substantial G-factor differences, which has even promoted speculation about G-driven MFEs in the geomagnetic field, 269. Contrary to the established paradigm, in principle, HFCs are not always needed to mediate the effects of weaker MFs on RP and similar reactions. In immobilized systems, the symmetry-breaking effect needed for ST interconversion can be provided by the electron-electron dipolar coupling, 270. This effect manifests in systems of more than 3 interacting radicals, whereby the slow decay of the dipolar interaction with distance permits spin-selective RP recombination in the presence of a remote, inert bystander radical. Although not yet experimentally verified, model calculations suggest that this effect could be relevant for the recombination of peroxyl radicals in the oxidative degradation of lipid membranes, 217, and that it might enhance the directional sensitivity of the cryptochrome-based inclination compass, 271. Even larger enhancements to MFEs on cryptochromes have been predicted if the third radical is reactive and also undergoes a spin-selective recombination reaction with one of the radicals of the primary pair, the chemical anti-Zeno effect, 272,273. 4.4. What is the likely influence of inter-radical interactions? So far, we have considered interactions between the unpaired electron spins and the effective local MF, but what of the interactions between the unpaired electron spin themselves? Taking the example once again of RP, the S and T spin states are energetically separated by the exchange interaction J-R, which is often assumed to depend exponentially on the radical separation. 225, Figure 6, D. When the radicals are close to one another, Jr can be very large, and the energy difference between the s and t spin states is such that their mixing cannot be efficiently driven by the comparably weak HFCs. To realize sensitivity to weak MFS, it is therefore necessary for the radicals to become separated in some way for J, R, to be overcome and for S, T interconversion to occur. The same principle applies to the electron-electron dipolar interaction, D, which is typically weaker than Jr, but decays less dramatically with distance, with the inverse third power rather than exponentially. When a paramagnetic molecule can freely diffuse in a biological system, one can imagine how spin-correlated pairs might separate and subsequently re-encounter on a timescale that allows spin-state evolution to occur before the system relaxes to equilibrium. Diffusive separation is not possible for radical intermediates that are to some extent fixed within the same rigid structure, for example, in a protein. It is here where the effect from weak MFS on many biological RP systems becomes more challenging. Radical separation in love domains, 274, and some B12-dependent enzymes, 275,276, for example, are too small, 1 nanometer, for Jr not to dominate. In these examples, the system would be forced into either the S or T state, and ST interconversions by the comparable weak HFCs abolished. Some B12-dependent enzymes, such as ethanolamine ammonialyase, EAL, however, show evidence of radical separations that might be sufficient. Nanometer for spin-state mixing to occur, 277, especially when the large difference in HFC for the B12RP is taken into account, 255. The suppressive effect of interradical interactions, J, R, and D alike, poses a particular challenge to cryptochrome-based magnetoreception. Theory suggests that, for a 50 UTMF to have a strong impact, the RP should be separated by 3.5 nanometers or more, 278. In an attempt to address this issue, it has been suggested that a matched exchange and electron-electron dipolar interaction, J-D compensation, could partly restore the S-T degeneracy that facilitates significant low field sensitivity, 278. Where J-R had been experimentally determined, however, it was found to be too small in absolute value to realize the required matching condition, 279, and theoretical studies suggest that J-R decompensation is likely to be ineffective in cryptochrome RPs, 271. It might therefore appear that, in proteins, maximal magnetic sensitivity would result in radicals being separated as far as possible. Yet the RP must also undergo a spin-selective recombination process within its coherent lifetime, figure 6e. Animal cryptochromes can undergo efficient electron transfer to FAD along 4 tryptophan residues, 280, while the RP involving the 4th tryptophan is better. Quantum Rep, 2021, 320. Separated, 2 nanometers, 281,282. A small but significant MFE by MT strength fields seems only to originate from the transient RP involving the 3rd tryptophan, 259. The 4th tryptophan is too distant from the FAD for significant recombination within the coherence lifetime, 283. In the presence of a significant exchange interaction that is J, R, 6 equal to 0, the S can be degenerate in 0th order with either the T+1 or T1 state at a particular value of the applied MF. 225 is illustrated in figure 6c, black plot. Here, magnetic sensitivity can only ensue if the system is sufficiently immobile to permit significant hyperfine-driven spin-state conversion at the level crossing configuration. The MF at this point, however, is usually large compared to those typically encountered environmentally, and this mechanism is therefore unlikely to facilitate low-field MFEs. 4.5. Are spin-selective reactions widespread in biology? Even if the conditions are amenable for a spin-correlated system in biology to undergo spin-state interconversion in the presence of AMF, a MFA will only manifest if the reaction is in some way spin-selective. A case in point is the ADO-CBL-dependent enzyme, EAL. As discussed in the sections above, the reaction dynamics of the COB2 alumin adenosyl RP are magnetically sensitive if generated photolytically 242,255,256,284. The same RP is produced on substrate binding to eel, and the RP separation and difference in HFCs could both be sufficient for spin-state mixing to occur. 277. Yet, the eel-catalyzed reaction is not impacted by exposure to MFS of a similar magnitude to those that impact B12 photochemistry. The homolysis step in eel is kinetically coupled to subsequent abstraction from the substrate by the adenosyl radical, which outruns the spin-selective recombination. Thus, recombination is minimal, and no MFA is observable. 285. An example that one might expect to be similar to eel is DNA photolyase. MFAs have been observed on the yield of a flavin tryptophan RP in photolyase in the absence of DNA and in the presence of potassium ferricyanide. 286. Ferricyanide oxidizes the FAD so that the radical photochemistry proceeds in a similar fashion to the photoreduction of cryptochrome. When repairing DNA, by contrast, fully reduced FAD is excited, resulting in electron transfer to DNA, generating a spin-correlated RP. DNA repair and RP recombination in photolyase is usually complete within 1 nanosecond, 287 however, and it therefore seems unlikely that the spin state has time to evolve before recombination. Such a reaction is thus unlikely to be spin-selective, and it was therefore greeted with some surprise, 288, when MFEs were nonetheless reported for DNA repair by photolyase, measured using a novel electrochemical setup, 289. Thus, a coherently interconverting spin-correlated RP is not always enough for a biologically meaningful MFE to manifest. Even if a reaction is spin-selective, however, it must to some extent be rate-limiting to the biological outcome. As discussed above, the mechanism of the enzyme cytochrome P450% reductase, CPR, proceeds via at least one spin-correlated RP generated by electron transfer. Again, however, it is an example of a biological system with RP intermediates whose reaction is not magnetically sensitive, 290. It has been demonstrated that electron transfer in CPR is gated by protein motions, 237, which occur on a much slower timescale than the relevant spin dynamics, and therefore spin selection does not dominate the catalytic kinetics. 4.6. Conclusions and outlook. T-power lines, domestic appliances and medical imaging equipment are now ubiquitous. Despite this, and the fact that both paramagnetic molecules and sources of spin correlation appear to be commonplace in biological systems, robust observations of biological MFAs remain rare. As we have discussed here, there are stringent requirements that must be fulfilled to elicit magnetic sensitivity on spin-selective reactions, Figure 6. The likelihood of all the necessary conditions being met in any one biological system appears to be low and are perhaps easier to realize in diffusing systems than immobilized radicals. While accidental magnetic sensitivity cannot be ruled out per se, it seems possible. Quantum Rep. 2021, 3:21, that protein magnetoreceptors, should they truly exist and be physiologically relevant, might need to have been evolutionally optimized for large MFAs to manifest. Arguably, the prime candidate for biological MF sensitivity is cryptochrome. Challenges remain even here, however. Open questions include the actual identity of the magnetically sensitive RP, no less, how the system might overcome interradical interactions, and the spooky effects caused by radiofrequency MFS, for example, 291,292, that have such tiny amplitudes so as to suggest unimaginably long coherence lifetimes, 293,294. Thus, our current understanding is incomplete at best. What we have gleaned so far, however, is enticing and remains consistent with the ability of some animals to perceive the Earth's MF by using a mechanism that to some extent is underpinned by non-trivial quantum effects, 224,295. In principle, it is a potentially powerful tool which could, for example, allow coherent control of physiological reactions. As for the question of whether the constraints are insurmountable, there is still a long way to go. 5. Proton tunneling in DNA. 5.1. DNA replication. All cellular genes are encoded as strings of nucleotides of the bases guanine, G, cytosine, C, adenine, A, and thymine, T, within DNA molecules. Each DNA molecule consists of two complementary strands in which A is paired with T and C with G, forming the famous double helix. During DNA replication, the strands are separated and the enzyme, DNA polymerase, synthesizes a complementary copy of each parent strand. The substrates for the enzyme are deoxynucleoside triphosphates, DNTPs, which are polymerized using the parental strand as template, sequentially assembling the corresponding nucleotide bases, one at a time, to the growing strand. The complementarity between the DNA bases and its essential role in replication was first proposed by Watson and Crick in their seminal 1953 paper, 296, where they suggested that the double helix structure is held together by specific interactions between pairs of bases. These canonical AT and GC Watson-Crick WC base pairs have a characteristic WC geometry, whereas the wobble GT, the wobble AC, and most other types of mismatches distort this geometry, as depicted in Figure 7. The fidelity of replication depends primarily on the ability of polymerases to select and incorporate the complementary base. However, any one of the four possible bases may arrive at the active site because, as far as we know, access to the active site by the DNTPs is governed only by diffusion. The task of identifying the unique complementary base for each template base among the various alternatives and assembling them in the order specified by the template strand can be described as an unstructured database search. Grover's quantum search algorithm is the optimal solution for this kind of problem, and its remarkable properties have motivated the proposal that quantum dynamics may be involved in DNA replication. 297. This possibility is allowed since the compatibility of replication and self-reproduction with the unitary laws of quantum mechanics and the no-cloning theorem has been demonstrated by the constructor theory of life. 298. Quantum Rep. 2021. 3. Self 22. Figure 7-7. The WC geometry of the canonical. Eta tandem G. CGC base pairs. Left, compared at the theta distorted. Geometry of the wobble G of spontaneous mismatches right. Geometry and A. Right, 5.2 spontaneous mutagenesis. Their area, many environmental and endogenous factors that can compromise the thean integrity of farf the genome induce mutations during replication, including of light, UV light, ionizing I9 ionization radiation, and reactive oxygens, PCS, 299. 299. In the final section of that semerth, that 1953 1953 paper, 296, Watson and Crick proposed a more subtle mechanism for spontaneous mutations that relied only on the intrinsic properties of the nucleotide bases. They hypothesized that since each base can exist in, at least, two readily interconvertible tautomeric forms, A, C, and G, mismatches could occur if one of the bases adopts its rare and energetically less favorable form, figure 8AB. Crucially, the geometry of the canonical AT and GC-WC base pairs is almost identical to the geometry of those tautomeric WC-like mismatches, which could thus be accommodated in the binding pocket of the active site of a DNA polymerase. The first experimental evidence consistent with this hypothesis was reported in 2011 when high-resolution X-ray crystallographic analysis demonstrated the presence of an AC mismatch in the active site of a DNA polymerase, 300. In that same year, a second study observed the crystal structure of an active polymerase poised to misincorporate G opposite a template 301. As predicted by Watson and Crick, Both studies observed WC-like mismatches that mimic the geometries of the canonical base pairs. Hence, this geometry mimicry, which has been confirmed in several subsequent studies, could enable the WC-like mismatches to evade the fidelity checkpoints and lead to spontaneous point mutations. 302. More recently, NMR relaxation dispersion has been successfully employed to visualize WC-like mismatches involving tautomeric base mismatches in both DNA and RNA duplexes and explored their dynamic equilibrium. 300 and 3304. The results suggest that rare tautomeric mismatches, despite their short lifetime and relatively low populations, might play an important and universal role in replication, transcription, and translation errors. Quantum Rep. 2021. 3 and 23. Figure 8. AB. Watson-Crick. WC. Hypothesis of spontaneous mutagenesis. WC like A, C, and G. Mismatches that preserve the geometry of canonical WC and lead to spontaneous point mutations. CE. Loudon's hypothesis of spontaneous mutagenesis. A double proton tunneling process converts canonical into tautomeric base pairs, which can be mutagenic when serving as a template in the next round of replication. The tautomerization process involves the relocation of protons, which are subatomic particles and obey the laws of quantum mechanics. In 1963, Per-Olof Lodin suggested that proton tunneling could facilitate the transition to the energetically less favorable tautomeric forms, 305. Specifically, he suggested that the canonical WC base pairs, AT and GC, could be converted to imino-tenol and genol-simino respectively, via double proton tunneling across the hydrogen bonds of each base pair, figure 8CD. If the two strands are separated whilst the DNA bases are in their tautomeric forms, then they could subsequently give rise to mutations when serving as a template in the next round of replication, figure 8E. Loudon's hypothesis has stimulated a vast number of computational studies and remains a very active area of research, as reviewed in the next section. Moreover, half a century after Loudon's hypothesis, our knowledge of how DNA replication is performed inside living cells has grown immensely. DNA polymerase is only one among tens of essential enzymes and other proteins involved in DNA replication that collectively form the replysome, 306. These include the DNA helicase enzyme, which unwinds the double helix by separating the two strands and could potentially separate the tautomeric bases envisaged by Loudon. However, a crucial point here is access of the solvent to the separated strands. If the single-stranded tautomeric base is exposed to the solvent, then it could exchange hydrogen atoms with the solvent to revert to the canonical form. In addition, Quantum Rep. 2021. 3. 24. This transition could even occur via an intramolecular proton transfer from one hydrogen bonding site to the other within the same nucleotide base, 307,308. However, tautomerization could result in a spontaneous point mutation if it occurs inside the active site of a DNA polymerase, since a mismatch would adopt a WC-like conformation that could mislead the polymerase to misincorporate it, as depicted in Figure 9. To examine this hypothesis, Lee et al. Studied the GT mismatch inside the active site of the low-fidelity human DNA polymerase A, Pol A, 309. Using a quantum mechanical, molecular mechanical, QM, MNM approach, the authors modeled the DNA pairing with QM while taking into consideration the influence of the broader biological environment of the active site of the polymerase with less accurate MNM calculations. Based on this approach, the free energy paths of the transfer process were found to be significantly influenced by the surrounding environment. Consistent with experimental data, these simulations indicated that the transition from wobble GT, WC like G-tenol, is stabilized by two catalytic residues of pol lambda. Furthermore, Maximoff et al. Conducted combined MD simulations and spectroscopic investigations to study the free energies of various conformations of GT mismatches within a mutant of pol A. They reported that the WC-like Janelle T mismatch dominates over the expected wobble GT conformation, 310. Therefore, these studies indicate that proton transfer in GT mismatches might take place inside the active sites of polymerases, yet it remains uncertain whether proton tunneling is involved. A positive indication that tautomerization might be facilitated by tunneling was provided recently by Rangadorai et al. 311. They extensively investigated the dynamics of the transition between a wobble and WC-like GT in duplex DNA by performing NMR relaxation dispersion in both water and D2O. The authors reported a kinetic isotope effect, Ki, as the rate of exchange between the two conformations of the mismatch was threefold slower in D2O than in H2O. This result provides the first experimental evidence in support of a transition state for tautomerization involving proton transfer. However, before the observed Ki can be attributed to proton tunneling, supplementary experiments will need to be conducted to measure its temperature dependence. 68, as elaborated above in section 2.2. Figure 9. Tautomerization leading from a wobble GT top to a WC-like genol T, bottom left, or G-tenol, bottom right. If either transition takes place inside the active site of a polymerase, the WC-like conformation could evade the fidelity checkpoints and result in a spontaneous point mutation. Quantum Rep. 2021, 3.53. In silico studies of proton tunneling, the energy landscape of the canonical amino-keto and tautomeric imino-enol forms of the AT and GC base pairs have been extensively modeled with ab initio quantum mechanical techniques such as density functional theory, DFT, 312 to 314, and post-Hartree-Fock approaches, 313,314. The reaction pathway of transfer can be modeled with transition state theory, 38, describing the double proton transfer process. The resulting double-well configuration, representing the canonical and tautomeric state, allows the proton to be transferred via quantum mechanical tunneling or classical over-the-barrier transfer. There have been numerous studies attempting to ascertain whether tautomerization can lead to non-trivial populations of mutagenic nucleotides. Broveretz et al. Have applied second-order moloplecit perturbation theory, MP2, with a high accuracy basis set, producing free energy curves that describe the proton transfer reaction, 315 to 318. Using the semi-classical Wigner correction, they calculated tunneling-corrected reaction rates and the average lifetimes of a wide range of tautomeric mismatches. It was found that when incorporating free energy contributions for the reactions of AT, imino-tenol, and GC-genol-cimino, the reverse reaction barriers become small, thereby leading to extremely short lifetimes, less than 0.1 Ps, of the tautomeric states, 319,320. Hence, based on their results, the specific reactions would be unlikely to be responsible for the generation of biologically relevant tautomers. While many ab initio simulations only involve a single base pair with no surrounding biological structure, some studies have attempted to capture the stacking interactions between neighboring base pairs. Villani, 321, suggested that the stacking interactions could enhance the hydrogen bond accepting capacity, depending on the hydrogen bond. Introduction of the stacking interaction leads to significant modifications of both static and dynamic properties of the GC base pair. 321. Stacking interactions alter the charge distribution and can thus modify the hydrogen bond strengths and proton transfer energy landscape. However, this model does not include other critical environmental influences, such as surrounding water molecules or the sugar backbone structure. Recent work by Solopolo et al. Tackles the full picture, employing a QM-M&M approach. 322. A base pair and a few nearby water molecules were included in the QM region, whereas the rest of the helix, water, and counterions were modelled with M&M. The authors reported that while the GC tautomer might be metastable in the gas phase, it becomes less unstable when in a B-form DNA configuration. The inclusion of environmental influences increases the free energy difference between the canonical and tautomeric state, thereby leading to a significant reduction of the reverse reaction barrier and the stability of the tautomeric state. Balancing the accuracy of the chemical calculations against the system size is a challenging problem. 323. In order to include a more massive biological structure, Solopolo et al. Adopted a computationally less expensive exchange correlation functional, BLIPE. However, such functionals, GGA or LDA, could decrease the accuracy of the description of DNA, while the level of theory used to model the base pairs could influence their structure and energies. At the same time, the combination of B3-LIP with an appropriately accurate basis set results in structures which are comparable to calculations with a much higher level of theory, MP2 perturbation theory calculations, 324 to 327, Godbeer et al., 328 to 330, combined DFT studies with the open quantum systems approach of using dissipative terms in the Lindblad equation and applied it to the AT base pair. They demonstrated that the dissipative Lindblad term couples the eigenstates localized in the canonical well to the excited states in the tautomeric well, thus leading to thermally assisted tunneling, whereby the environment couples to the system, providing it with a thermal boost. Furthermore, increasing the temperature of the bath strengthens the coupling to the environment and enhances the rate of thermally assisted tunneling. A similar enhancement can be observed in a simpler model by simulating successive von Neumann measurements, where an increase in the frequency of observation measurement leads to an anti-Zeno effect, 329. A transition state search using CASTEP at B3 ALIP level provided a quantum rep, 2021, 326, description of the potential surface for the proton transfer. Furthermore, Godbeer et al. Varied the potential shape, barrier, and asymmetry, and determined the effect on the tunneling probability, which was reported to have a maximum value on the order of 10 to 9 for the AT base pair. However, this study might have overestimated the reverse barrier of the reaction, resulting in a deeper second well corresponding to the tautomer, 315. Pohl et al. Used path integral molecular dynamics, corroborated by experiments of deuterium-induced NMR chemical shifts, to investigate hydrogen-bonded isocytosine pairs, an analogue of the GC pair, 331. They concluded that interbase proton transfer reactions across the hydrogen bonds do not take place in the isocytosine system, but it is unclear if this will translate directly to the GC pair. Path integral molecular dynamics were also employed by Feng et al. To investigate nuclear quantum effects, NQEs, directly in DNA base pairs, 332. Their results indicate that NQEs strengthen the duplex hydrogen bonding interaction by 0.5 kilocalories. Fang et al. Suggest that even at room temperature NQE are non-trivial. Further work is required to unpick the interplay between NQE strengthening hydrogen bonds and the stability of the tautomeric states. 5.4. Conclusions. The task of DNA replication is central and essential for all life. Quantum mechanical effects may be involved in the remarkable speed and efficiency with which this task is performed inside living organisms. On the other hand, quantum tunneling might lead to inaccurate replication, thus generating mutations that can drive evolution. Despite the remarkable progress that has been achieved in our theoretical understanding, experimental techniques and computational capabilities, additional efforts will be required before any conclusions can be drawn regarding these long-standing hypotheses. 6. Fluorescent protein as a novel model system for quantum biology. Both theoretical and experimental evidence has been accumulated, indicating that non-radiative excitation energy transfer mechanisms, including both coherent and incoherent energy transfer, play a fundamental role in a number of processes in biological molecules and biomolecular systems, such as in photosynthesis, 143 trillion, 148 billion, 150 million, 155,160. Non-radiative excitation energy transfer can be understood as an interaction mechanism, 333 to 336, mediated by coherent energy transfer that requires close proximity between donor and acceptor in order to preserve exciton coupling. It was therefore thought to be highly unlikely that coherent energy transfer takes place between fluorescent proteins, FPS, as fluorophore proximity is limited by the Fp beta barrel structure, and in a warm, wet biological environment, vibrational decoherence would rapidly intervene to obliterate quantum coherent molecular interactions. Nonetheless, evidence indicates that anomalous photophysical properties of Fp have been observed at room temperature, which cannot be understood explicitly within the boundaries of classic physics. 337 to 346. Surprisingly, recent experimental evidence observing generation of photonic entanglement in enhanced green fluorescent protein, EGFP, 339, and exciton coupling between dimeric yellow fluorescent proteins, Venus, 340, strongly supports the hypothesis that FPS can interact coherently under physiological conditions. In this section, we review the theoretical background of excitation energy transfer and describe recent experimental progress in the study of quantum effects in FPS. 6.1. Excitation energy transfer. In 1965, Forster proposed three main regimes for energy transfer between donor and acceptor fluorophores that share common atomic transitions: strong, intermediate weak, and very weak coupling, 336. This distinction is strongly correlated with the relative rates of energy transfer to the rate of excited state vibrational relaxation. A rate of energy transfer is defined as the reciprocal of the time required for the energy transfer from an excited state donor molecule to a ground state acceptor molecule. In the strong coupling regime, the rate of energy transfer is much larger than that of the vibrational relaxation. Quantum Rep. 2021, 327. Process so that all the vibronic subtransitions associated with the energy transfer in both molecules are resonant. When the interaction is approximated by the Coulombic coupling between absorption and emission dipoles, the rate that is efficiency of energy transfer is inversely proportional to the third power of the distance between donor and acceptor, R3. The strong coupling strength results in major alterations of the absorption spectrum and is thought to be possible to mediate excitation energy transfer up to 100 nanometers, 334. In the intermediate weak coupling regime, the rate of energy transfer is comparable to that of vibrational relaxation by an order of magnitude, in which coherent energy transfer is thought to prevail. The coupling strength between fluorophores within this regime is expected to lead to minor alterations in the absorption spectrum, such as Davidoff splitting of certain vibronic bands. In the very weak coupling regime, the coupling strength is much lower than the vibronic bandwidth, so that the vibrational relaxation takes place prior to energy transfer. In this regime, fluorophores act independently, so that it is expected that there is no alteration of the absorption spectrum. In addition, energy transfer is thought to be incoherent via FRET and is typically limited by a distance up to 10 nanometers. The interaction is inversely proportional to the 6th power of the distance between donor and acceptor, R3. 6. In 1974, Knox and Kenkre formalized the theory of slow dephasing coherent energy transfer based on the unification of the strong and weak coupling theories. 335. Figure 10 shows the rate of excitation transfer as a function of interaction energy as predicted by Forster for the strong, weak, and very weak coupling regimes, as well as predicted by the Kenkre and Knox formalism. Both Forster's theory and Kenkre-Knox's formalism hypothesized that review 28 energy transfer associated with the intermediate weak coupling regime should be coherent, but little is known about this regime experimentally. Figure 10.1. Zero comparison of transfer tests, rates predicted be Forster for, the four strong. The strong weak weakened, Verand weakfree regimes. Weak regimes and Thean formalism of Kenkre-Knoxto unity, Torth unity weak and weak strong and coupling strong theories. Coupling light Theo curves, upper and right scales, correspond to Forster's pairwise transfer rates as a function of interies. Light curves, upper and right scales, correspond to Forster's pairwise transfer rates as a function action energy, U, in which curves A, B, and C indicate Forster's strong, intermediate, weak, and of interaction energy, U, in which curves A, B, and C indicate Forster's strong, intermediate weak, very weak coupling regimes, respectively. The single function predicted by Kenkre and Knox is depicted and various weak, a heavy coupling curve, lower regimes, and respectively, left scales, the single parameters function alpha. Predicted and W indicate B, Kenkrather decay and Knox rate, and is depicted the ratios of energy heavy transfer curve, low respectively, and left scales, note. That the parameter supper, and dalfaretand, WS scalar syndicateer, takenth, decay into account rate, and in thea particular rate of energy case transfer, where respectively. Taken 8.8 1014 seconds 1 note was, that chosen D, upper adapt and right ref, scales 335, taken into account in a particular case where alpha equal to 1.8, 1014 seconds, 1 was chosen. Adapted from ref, 335. Fluorescent protein. Since the discovery of green fluorescent protein GFP isolated from the jellyfish Equoria victoria, 347, FP has become an essential tool to study modern cell biology by enabling genetic protein tagging and the visualization of cellular interactions in living cells, 348. Each FP chromophore is composed of 3 amino acids encased at the center of a cylinder-shaped, three-dimensional structure, which is the FP beta barrel. Many naturally occurring FPS tend to form dimers or tetramers, 349 to 351. Thanks to mutagenic studies, GFP can be genetically engineered to optimize its photophysical properties such as absorption emission spectra and brightness for bioimaging applications. For example, enhanced GFP, EGFP, 352, presents a higher fluorescence than that of wild-type GFP, and yellow fluorescent protein, YFP, emits light in the yellow spectrum, 353. Both EGFP and YFP are derivatives from wild-type GFP. Modification of the sequences allow further improvement of the photophysical properties of FP so that FPS are used to perform measurements of intracellular compounds and expression levels, 354 to 357. Nascent FPs are colorless, and so must undergo maturation to become fluorescent. The maturation process involves the folding of the beta barrel, torsional rearrangements, cyclization, oxidation, and dehydration of the chromophore, 358. For example, the chromophore of GFP is formed by the autocatalytical post-translational modification of 3 amino acids, Sus65 to 66-gly67, 352, and is embedded in the beta barrel that has a diameter of 24A and a height of 42, a 359. The B barrel is thought to play a role as a molecular bumper by isolating the fluorophore from molecular collisions caused by the external vibrational environment, 360. Thus, FPS are less accessible to external quenchers whilst interacting actively with their biological environment. Within the EGFP beta barrel, there is a cavity harboring 4 additional water molecules, 352. This interaction with the medium is necessary for proper function in terms of FP fluorescence, as dried films of EGFP showed a decreased fluorescence to third of the peak value of the hydrated protein, 356. The beta barrel is also thought to be implicated in limiting the proximity between adjacent fluorophores and to play a role to avoid self-quenching, 356. Structure-guided evolution of cyan fluorescent protein, CFP, leads to the creation of mu-turquoise E2. This mutagenic study, in conjunction with X-ray crystallography, provides evidence that modification of the beta barrel affects the brightness, maturation rate, photostability, fluorescence lifetime, and even the quantum yield, 361. 6.3. Forster resonance energy transfer. FRET is a non-radiative energy transfer mechanism mediated by Coulombic dipole-dipole coupling, by which excitation energy is transferred from an excited state donor molecule to a ground state acceptor molecule. Upon excitation, FRET occurs at close proximity, typically a range of 110 nanometers, with spectral overlap and appropriate relative dipole orientation between the donor and the acceptor fluorophores. 334. When donor and acceptor fluorophores have common atomic transitions, then energy transfer proceeds discretely via the FRET mechanism. Applications of FRET spectroscopy and microscopy in life science have rapidly increased over the past 2 decades because of the usefulness of FRET for proximity detection. 334,362. The extensive use of FPS for FRET measurements, 362,363, as a spectroscopic ruler, 364, has assumed that energy transfer between fluorophores takes place via an incoherent FRET mechanism, 333,365. This assumption is based on the understanding that coherent energy transfer between adjacent fluorophores will be prevented at the distances between donor and acceptor limited by the FP-B barrel structure. Moreover, physiological conditions are thought to promote rapid vibrational decoherence, thereby causing incoherent energy transfer, 334, in which fluorophores behave independently, and excitation energy is transferred discretely from a donor to an acceptor. However, it has been observed that FPS show anomalous photophysical behavior that cannot be explicitly explained by the theory of the very weak coupling, 337 to 346. Suggesting the possibility that excitation energy can be transferred coherently between FPS, even under physiological conditions. Quantum Rep. 2021, 3, 29, 6.4. Anomalous photophysical properties of fluorescent proteins. Schinelli et al. Reported in 2001 that at room temperature, the non-stationary dynamics associated with vibronic wave packets in GFP involved a dephasing time of 0.4, 337, which is 50 times slower than organic fluorophores, 336. In this study, an oscillatory modulation of the differential optical transmissivity after femtosecond laser excitation was observed and used to characterize the coherent dynamics of single-electron vibronic wave packets in GFP 337. In 2005, Young et al. Utilized polarization transient absorption spectroscopy to observe the rate of energy transfer among YFP dimers, and reported a 2.2 ps anisotropy decay constant, which corresponds to a bidirectional energy flow with a time constant of 4.4 ps 338, which is a surprisingly fast time considering a lifetime of YFP, 3 nanoseconds, 366. According to the formalism of Knox and Kenkre, 335, these results may be attributed to coherent energy transfer between FPS associated with the intermediate weak coupling regime. Circular dichroism, CD, absorption spectroscopy is one of the most powerful techniques for stereochemical analysis. CD is the difference between the absorption of left and right, circularly polarized light, and can be used to identify the position of electronic bands and study the electronic coupling associated with the strong and intermediate coupling regimes, manifested by Davidoff splitting, 367 to 369. Visser et al. Studied CD spectra from blue fluorescent protein BFP, CFP, EGFP, and YFP, which are variants of GFP and tetrameric discosoma S-pore red fluorescent protein, DS-red, 343. In this study, DS-red presented a CD spectrum with an excitonic signature at the inflection point of the low wavelength edge of the absorption band, whilst the other FPS CD spectra coincided with their absorbance spectra. These results suggest stronger than expected coupling between the chromophores in DS-red, 343. Note that DS-red oligomerization to form tetramers containing 4 chromophores occurs even at low concentrations. Photon antibunching was detected using a scanning confocal microscope with single molecule sensitivity, indicating that, upon excitation, single chromophore emits photons one at a time, rather than, as might be expected for an entirely classical system, the 4 chromophores emitting photons independently of each other, 342. In 2009, Kushik et al. Performed FRET measurements using multiple FRET acceptors and consecutively observed anomalous surplus energy transfer. In this study, predictions based on a FRET model underestimated the experimentally observed energy transfer efficiency. The authors concluded that either the theoretical assumptions of the FRET model are incorrect for this system, or additional energy transfer pathways were active. 344. 6.5. Generation of polarization entangled photon pairs in an ensemble of EGFP molecules. Thanks to recent advances in techniques for optical spectroscopy, it has become possible to precisely manipulate and monitor light for the study of light-matter interactions in biological systems. In 2016, Shi et al. Reported that, unlike organic fluorophores, EGFP is less affected by background noise in response to the generation of spontaneous four-wave mixing, SFWM. In this study, EGFP demonstrated a coincidence-to-accidental ratio, CAR, of 145, which is approximately 10 times larger than that of the best-performing organic fluorophore, pyrimethene 556, whose CAR is 15 chapter 346. In 2017, the same group demonstrated the generation of polarization entangled photonic states from the SFWM process in an ensemble of EGFP molecules at room temperature through 2 horizontally and 2 vertical polarized pump pulses, 339. The data were used to reconstruct the density matrix of the entangled photonic states. The result indicates that EGFP fluorophores are relatively insensitive to environment thermal decoherence, which may be attributed to the isolation of an encapsulated fluorophore inside the FP beta barrel structure, 360. 6.6. Exciton coupling in homodimers of yellow fluorescent protein Venus 206. In 2019, further experimental evidence for room temperature coherent molecular interactions between FP fluorophores was obtained by Kim and others. The study reported unexpected FP quantum behavior resulting from excitonic coupling in homodimers of dimeric. Quantum Rep. 2021 3:30. Yellow fluorescent protein Venusat 206 under physiological conditions, figure 11a, 340. Time-resolved fluorescence anisotropy measurements were used to reveal ultra-fast energy transfer, defined as faster than the instrument response function of the anisotropy system, approximately 146 Ps, between FP fluorophores when Venusat 206 FPS dimerize, figure 11b. CD absorption spectroscopy revealed Davidov splitting, figure 11c, in Venusa 206 dimers, which is a quantum mechanical manifestation of fluorophore excitonic coupling. Moreover, a novel approach combining photon antibunching, AB, with fluorescence correlation spectroscopy, FCS, AB-FCS fingerprinting, was used to compare the number of independent emitters and fluorescent fluctuating molecules in the same sample. Figure 11d shows the results of the AB-FCS fingerprints for monomeric Venusa 206K, black, monomeric Venusa 206K tandem dimer with the Venus fluorophores separated by a 20 nanometers linker, Venusa 206K to 20 nmtd, green, and dimeric Venusa 206 tandem dimer, Venusa 206 TD, red, which indicate that the Venusa 206 TD behaves as a single quantum entity. Alteration of Venusa 206 TD emission behavior as a result of exciton coupling between FP fluorophores again provides review strong evidence for quantum coherent molecular interactions. Note that singlet 31 annihilation, which is an alternative classical description for reducing 2 excitation events to 1 emission event and is supposed to be dependent on excitation power, cannot be employed power. Cannot explain be employed strong tope explainin Venusa 2. 106 TD strong AB, in since a Venusa 206 TDA dependence since a dependence on excitation and power, waste excitation not observed. Power was it, is not also observed, not worthy it, is also noteworthy ifs, in case that is singlefs, in case fluorophoria, single within fluoro, beta barrel structure 4, within that arabeta barrel easily structure modified that BR neti, Sealy engineering modified by techniques. Genetic engineering making tech, them ideal neeks. making them ideal tools, as well as novel model systems, for studies of quantum tools, as well as novel model systems for studies of quantum effects in a biological system. Effects in a biological system. ABD Figure 11. Quantum Leven, Quantum Signature Subserved Observed Dinin Homodimers, yellow fluorescent protein Venusat, 106 Venus Ard, 106, when they dimerize. A. An illustration of a pair of dimeric Venusat 106 molecules based on the crystal structure. Venus chromophore, and its B. A. An illustration of a pair of dimeric Venus 106 molecules based on the crystal structure. Venus chromophore and its barrel are depicted as blue spheres and yellow ribbon structures, respectively. A residual, alanine 206, that involves in B barrel dimerizational, depicted as blue spheres red. The and separation yellow proximity ribbon structures between center to centre respectively. FPA fluor 04 res residual, alanine 206, in crystals is that 2.5 involves in dimerization, and the closestis depicted distances between red. Theat separation fluor 4 res proximities 1.5 nanometers between B time resolved center to center fluorescence fluorophorizani sotropion of crystals Venusat 106 chaos 2.5 nanometers, and the black closest monomeric distance Venusat 206 K to 20 nmtd between its fluorophores, green, and is 1.5 Venus 206 tdnm. B. Time resolved red, C. Dimerization fluorescence celtas, its anisotropy circular Dicra ismuf Venusa 206 K spectrum that results in Davidov splitting. For each fluorophore concentration, the Venusat 206 CD absorption spectrum, black, monomeric Venusat 206K to 20 nmtd, green, and Venusar 106 TD, red. C. Dimerization alters its circular dichroism was subtracted from its matching CD spectrum of Venusa 206KD, ABFCS fingerprints for the 3 samples. The inset spectrum shows that results Santibunchingine Davidov measurement splitting. O for Venus 206 Keach. Fluorophore, black. Venusar 206 TD concentration, red. Thean Venusar 206, Venusar 206K to 20 CDNM, TD absorption, green, spectrum. Was subtracted adapted from ITS 340. Matching. CD spectrum of Venusa 206KD, ABE FCS fingerprints for the 3 samples. The inset shows the anti-bunching measurements of Venusa 206K, black, Venusa 206, TD, red, and Venusa 206K 20 nanometers, TD, green. Adapted from Ref. 340. 6.7. Conclusions and Outlook. Quantum Rep. 2021, 331, 67. Conclusions and Outlook. It is apparent that the results of the generation of photonic entanglement in EGFP 339 and excitonic coupling between dimeric Venus 206 FPs 340 at room temperature support the hypothesis that FPs have evolved a mechanism to prolong quantum coherence under physiological conditions and might therefore have unique photophysical properties. FP is a simple, accessible, and amenable system that can be genetically manipulated. Moreover, a unique advantage of FPS over other probes, such as organic dyes, is that they are resistant to the presence of external quenchers, 360, as well as presenting a high quantum efficacy, 370. Application of these approaches will provide an insight into understanding how biological systems maintain quantum effects in a warm, wet, and complex biological environment, as well as how such quantum effects influence biological processes and behaviors. Furthermore, knowledge learned from these approaches may open a new avenue towards quantum bio-inspired technologies, such as the development of efficient quantum light sources and next-generation biosensors that exploit quantum behavior. 7. Quantum coherence in neuronal ion channels. At first sight, it does seem unlikely that delicate quantum effects, such as coherence, tunneling, entanglement, or spin, could play significant roles in a warm, wet brain. However, the Nobel Prize-winning UK mathematician Roger Penrose, together with the American anaesthetist Stuart Hameroff, made probably the most audacious claim for quantum biology in recent years in their proposal that quantum coherence in neuronal microtubules is capable of quantum computing and is the substrate for consciousness. 371,372. This proposal has generated a great deal of discussion and criticism and it is fair to say that it has not received significant support in either the physics or neuroscience community, and so will not be considered further in this review. The interested reader may consult several recent accounts of the theory, for example, 373. In this review, we will focus instead on what we believe is a much more likely substrate for quantum mechanical effects in the brain: neuronal ion channels. Neuronal ion channels play a fundamental role in nerve, and thereby brain, function. With pores only around 0.1 nanometers diameter, Figure 12, small enough to ensure that ions travel in single file through the channels, they are of a scale that might conceivably exhibit quantum phenomena. A resting neuron maintains a voltage difference across its membrane of about 70 MV, negative on cytoplasmic side of the membrane. This is ensured by the activity of sodium plus K plus pumps situated in the plasma membrane of the cell, along with the different permeabilities of the membrane itself to these ions. On receipt of a neurotransmitter from an upstream cell, a neuron will open or close ion channels that either increase or decrease the membrane voltage. If a neuronal membrane becomes sufficiently depolarized to about 55 MV, then further voltage-gated ion channels in the neuronal membrane respond by opening or closing, causing a wave of depolarization that travels along the nerve axon to its terminus, where it triggers the release of neurotransmitters that stimulate a downstream target. Voltage-gated ion channels, VGCs, are highly conserved transmembrane proteins composed of subunits arranged together in a tunnel-like structure that provides a hydrophilic channel through which ions can flow depending on their electrochemical gradient. 374. Quantum Rep. 2021, 332. Figure 12. The figure structure 12. The of structure the bacterial of the potassium bacterial canal potassium xa channel with its KCSA4 transmembrane with its 4 transmembrane subunits. Left subunits and the selectivity filter, right. Left, the and selectivity. Selectivity filter is composed filter, right, of. The 4 selectivity amino acid filter chains is composed on each from of 4 1-aminoof thiacid 4 chains, protein subunits, 1 each from one of the 4 protein subunits constituting the ion channel. Each chain is made up constituting the ion channel. Each chain is made up of 5 amino acids, from which a carbonyl group oxygen atom is of 5 amino acids, from which a carbonyl group oxygen atom is orientated towards the pore. Orientated towards the pore. Adapted from Ref. 375. Adapted from Ref. 375. Properties central to the functionality of VGCs are ion selectivity and ion conduction. Ion selectivity is conferred by a portion of the protein known as the selectivity filter, Figure 12, that discriminates between different ions based on their diameter of a few angstroms and charge. Ions passing through the channel dispose of their hydration shell before entering the pore, so they travel through the channel single file. Inside the selectivity filter, highly conserved motifs of charged residues pull the ions within the channel due to the attractive force of Coulomb interactions. 376,377. Voltage change in the membrane switch the channel between its open and closed state via long-range protein motions in a specialized segment known as the voltage sensing domain. 378. Ion channels, such as the well-studied bacterial KCSA potassium channel, exhibit several exceptional features that suggest the possibility of quantum mechanical enhancement. The channel combines a high ion throughput rate of 108 ions, which is close to the diffusion limit, combined with an efficiency of 104, one for discriminating between potassium and sodium ions. This is even more remarkable since the sodium ion is smaller than the potassium ion and would thereby be expected to pass easily, though the potassium ion channels 376,379 to 381. It has been argued that the noteworthy values of selectivity and conduction are hard to account for through classical mechanisms, particularly as VGC proteins are flexible and thereby subject to thermal fluctuations. 382. Moreover, several dissimilarities occur between potential barrier heights described by the laws of thermodynamics and ion translocation rates empirically observed within channel proteins. 383. Quantum mechanics has thereby been enlisted as a potential contributor to ion channel function in order to account for their exceptional properties. The key concept that has been applied to ion channel conduction is quantum coherence, QC, the coexistence of different non-exclusive quasi-classical descriptions of the same physical system, definable as the probability distribution for the sum of each possible measurement on that system, that is particle, due to its wave-like nature. 384. QC is usually an extremely short-living phenomenon, rarely lasting more than a few femtoseconds at ambient temperatures, due to rapid decoherence. 4. However, recent calculations have significantly increased the pre-existing times for QCs in neurons, identifying ion channels as preferential sites for the research of non-trivial quantum effects in the CNS. 385. Specifically, theoretical approaches have indicated that short coherences may be not just beneficial, but also necessary to explain the fast-directed permeation of ions through the potential barrier of the filter. 375,386. Quantum Rep. 2021. 333. In one recent study, it was demonstrated that a distance of 0.3 nanometers between two ions within the ion channel selectivity filter allows the ions to be described in a state of quantum superposition. 383, and thereby consistent with wave-like propagation of ions through the channel. In this scenario, decoherence may promote the energetic transition of the transiting ion between the different binding sites of the selectivity filter, allowing its wave function to spread along the electric landscape, represented by the charged residues within the selectivity filter, with decoherence enforcing the directionality of ion transport. 383. Consistent with this theory, simulation studies revealed dynamics closest to those experimentally observed when the system was considered to be neither fully coherent nor completely classical, but oscillating between both states. 375,386. In another study, incorporation of quantum interference terms in the initiation of nerve firing replicated the characteristic fast onset of action potentials measured by electrophysiological recordings of cortical neurons. 387. More recently, the potential for quantum tunneling of ions through closed channels has been theoretically investigated. In one study, the Goldman-Hodgkin-Katz (GHK) equation for membrane potential was implemented to investigate the contribution of quantum tunneling in channel conductivity. 388. The investigation found significant probabilities for tunneling of several ions, such as lithium and potassium, prompting the proposal of a quantum electrochemical equilibrium model for ion conduction, and a potential explanation for the well-known, but currently unexplained, therapeutic depolarizing effect of lithium in the bipolar disorder. 389,390, and the ion dynamics of potassium in the phantom limb pain. 391. The concept of a quantum-confined superfluid, defined by Wen and colleagues. 392, in 2018, has also been adapted to propose a quantum-confined ion superfluid model of neuronal conduction that aims to account for the near-instantaneous response characteristic of nerve signal transmission. 393. The very short lifetime of QC in nerves likely restricts the potential role of non-trivial quantum effects in neurophysiology. 385. However, according to a recent proposal, The effects of even short-lived QC could be amplified by electromagnetic fields generated by neuronal electrical activity. 10. Extracellular electromagnetic fields are known to influence neural firing in the process known as ephaptic coupling and have been implicated in neuronal synchronization and gap junction transmission. 394 to 396. Recent studies suggest that the complex nonlinearity of brain dynamics may amplify small, even quantum-level fluctuations. 397,398. Possibly mediated by EM field influences, 394,399. In conclusion, although many interesting theoretical studies suggest that quantum mechanical phenomena such as coherence and tunneling play a role in ion channel conduction and thereby brain function, experimental studies lag behind the theoretical work and much needs to be done to establish a quantum biological neuroscience. 8. Conclusions. It is clear that quantum biology is a growing area of interdisciplinary research with contributions from quantum physicists, molecular biologists, biochemists, mathematicians, engineers, chemists, and spectroscopists. As described here, important advances have been made in the well-established fields such as photosynthesis, enzyme catalysis, and avian navigation, as well as in less established areas such as fluorescent proteins, ion channels and proton tunneling in DNA. Yet questions and challenges remain. Although there is no doubt that the molecular machinery of life involves the motion of individual electrons or protons that necessarily involves quantum phenomena such as coherence, tunneling, or entanglement, establishing a biological function for these quantum features remains a challenge. For obvious reasons, most experimental data have been obtained from in vitro systems capable of generating measurable signals. However, even the best available in vitro systems, such as photosynthetic complexes and whole enzymes, consist of thousands or millions of atoms that are already too complex to realistically model at the quantum level. One approach to address this problem, as outlined in this review, is to utilize nanotechnology approaches to build biomimetic systems that are simpler than their biological. Quantum Rep. 2021, 334. Counterparts and thereby more amenable to mathematical modeling and computation. This is clearly a fruitful area for further development and even technological applications. Nevertheless, in vitro systems, even biomimetic ones, are unlikely to convince the skeptics that quantum biology is relevant to life. Proof requires demonstration that quantum phenomena play a role in biological function in living cells and thereby contribute to fitness. Yet, quantum biological phenomena have only so far been investigated in a limited range of organisms, most of which, such as birds or photosynthetic bacteria, are relatively intractable to the powerful tools of molecular biology and synthetic biology. The challenge of the future will be to transfer measurable quantum biological systems into the more tractable workhorses of molecular biology and synthetic biology, such as E. coli or yeast, to engineer systems that allow simultaneous detection of quantum-level events and their macroscopic outcomes. This challenge will require a new generation of scientists who can work across the disciplines of physics, chemistry, and biology. This is not a new challenge. The discipline of biochemistry necessitated a similar merging of chemistry with biology about 100 years ago. It required the training of a new generation of students and researchers able to take on the challenge of working at an interdisciplinary frontier. The fusion of biochemistry with physics to build the new discipline of quantum biology will be an even harder challenge, yet one that is the principal aim of the Leverhulme Quantum Biology Doctoral Training Centre, QBDTC, at the University of Surrey. Our centre currently trains 20 PhD students, many of whom contributed to this review, with the skills to work across this hugely interdisciplinary field. With talented researchers enthused by an exciting new field, quantum biology has a great future. Author contributions: JAK and JM conceived the study. All authors discussed and wrote the manuscript. LOJ, DJH, AISH and NSS contributed Section 2, Quantum Tunneling and the Importance of Protein Dynamics in Enzymatic H-Transfer Reactions. ETN, MGS, SRPs, and GSE contributed Section 3, quantum effects in photosynthesis. EMD, DRK and RJ contributed Section 4, magnetic field effects on spin-dependent reactions in biology. LS, AP, EMS, JAK, SMH, W and JM contributed Section 5, proton tunneling in DNA, as PJ and YK contributed Section 6, fluorescent protein as a novel model system for quantum biology. FB, RL, SRPs, JM and KJ contributed Section 7, quantum coherence in neuronal ion channels. All authors have read and agreed to the published version of the manuscript. Funding. Contributions from all authors within the Leverhulme Quantum Biology Doctoral Training Centre at the University of Surrey were funded by a Leverhulme Trust Doctoral Training Centre grant number DS 2017-079. FB is funded by the Astor Foundation. Institutional Review Board Statement, not applicable. Informed Consent Statement, not applicable. Data Availability Statement, not applicable. Conflicts of Interest. The authors declare no conflict of interest. References. 1. McFadden J. Al-Khalili, J. The Origins of Quantum Biology. Proc. R. Soc. A 2018, 474, 20,180,674. Cross-ref. PubMed. Schrödinger, E. What is Life? Cambridge University Press, Cambridge, UK, 1944. 3. Melkik Av, Krennikov, A Non-Trivial Quantum and Quantum-Like Effects in Biosystems, Unsolved Questions and Paradoxes. Prog. Biofees, Mol. Biol, 2015, 119, 137-161. Cross-Reef. Tegmark, M. Importance of in Brain Processes. Phys. Reverend E. 2061, 4194. Cross-Reef, PubMed. Quantum Decoherence. 5. Mac Chemish L.K., Reimers, J.R., Mac Kenzie, R.H., Mark, A&E, Hush, N.S. Penrose Hameroff Orchestrated Objective Reduction Proposal for Human Consciousness is not biologically feasible. Phys. Reverend E. 2009, 80, 021912. Cross-Ref. Dwan H.G., Prokorenko, the 6, Cogdell R.J., Ashraf K., Stevens Al., Thorwart M., Miller R&D. Nature does not rely on long-lived electronic quantum coherence for photosynthetic energy transfer. Proc. Natal, Akad Shai, USA 2017, 114, 8,493 to 8,498. Cross-Reef. Wilkins, D.M., Datani, N.S., Why Quantum Coherence is Not Important in the Fenner-Matthews-Olson Complex. J. Chem, Theory Comput. 2015, 11, 3,411 to 3,419. Cross-Reef. 8. Lambert, N. Chen, Y.N. Chang, Y.C. Lai, C.M. Chen, G.Y. Nori, F. Quantum Biology. Nat, Phys. 2013, 9, 10-18. Cross-Reef. Quantum Rep. 2021, 3:35. Mosini M., Omar E. Engel, G.S., Plenio, M.B. Quantum Effects in Biology, Cambridge University Press, Cambridge, UK, 2014. McFadden J. Al-Khalili, J. Life on the Edge, The Coming of Age of Quantum Biology, Broadway Books, Portland, or USA, 2016, 10-12. Scholz G.D., Fleming G.R., Chen L.X., Aspuru Guzik A., Buchleitner A., Kokodiev, Engel, G.S., Van Grondel R., Ishizaki A., Jonas D.M., Using Coherence to Enhance Function in Chemical and Biophysical Systems. Nature 2017, 543, 647-656. Crossref. Brooks, J.C. Quantum Effects in Biology, Golden Rule in Enzymes, Olfaction, Photosynthesis, and Magnetodetection. Proc. R. Soc. A Math. Phys. Ang Sai. 2017, 473, 20,160,822. Cross-Reef. 13. Murray A., Adams B., Ringsmouth A.K., Ferretti M., Gruber J.M., Hendricks R., Schuld M., Smith S.L., Sinaisky the 1, Kruger T.P. The Future of Quantum Biology. JR SOC. Interface 2018, 15, 20,180,640. Crossref. 14. Barnson BJ, Colby TD, Chin, JK, Goldstein BM, Klinman JP. A link between protein structure and enzyme catalyzed hydrogen tunneling. Proc Natal, ACID CI, USA 1997, 94, 12,797 to 12,802. Cross Reef. Pisran J. Sutcliffe, MJ. Scrutton. NS enzymatic H transfer requires vibration-driven extreme tunneling. Biochemistry 1999, 38, 3,218 to 3,222. Cross Reef. PubMed, Alhambra C. Corchado J. Sanchez ML, Garcia Villoca M. Gao J. Trular, DG. Canonical variational theory for enzyme kinetics with the protein mean force and multidimensional quantum mechanical tunneling dynamics. Theory and application to liver alcohol dehydrogenase. J. Phys. Chem. B. 2001, 105, 11,326 to 11,340. Cross-ref. Billiter Senior, Webb, SP, Agarwal, PK, Iordanoff, T, Hams-Schiffer, S-hydride transfer in liver alcohol dehydrogenase, quantum dynamics, kinetic isotope effects, and role of enzyme motion. J. Am, Chem. ESOC, 2001, 123rd, 11,262 to 11,272. Crossreef. Klinman, J.P. Environmentally coupled hydrogen tunneling. Linking catalysis to dynamics. Ure, J. Biochem. 2002, 269, 3,113 to 3,121. Cross-ref. 19. RK evidence for environmentally coupled hydrogen tunneling during dihydrofolate reductase catalysis. J. Am Chem. ESOC. 2003, 125th, 13,372 to 13,373. Crossref.
Garcia Villoca M. JLJ. Carplus M. Trular DG. How enzymes work. Analysis by modern rate theory and computer simulations. Science 2004, 303, 186 to 195. Cross.
Liang Zex Klinman, JP. Structural bases of hydrogen tunneling in enzymes, progress and puzzles. Kerr, Open. Struct. Biol. 2004-14, 648 to 655. Crossreef. 22. Liang Zix, Li T, Rezinkar, An, Inji, Klinman. JP. Thermal activated protein mobility and its correlation with catalysis in thermophilic alcohol dehydrogenase. Proc. Natal, ACAD, C. USA 2004, 101, 9,556 to 9,561. Crossref. Mazgrau L, Rujanikova A, Johannesson, Lowe Hothi P, Basran J, Ranahan Key, Mulholland, AJ, Sutcliffe MJ, Scrutton, NS, Lees D. Atomic description of an enzyme reaction dominated by proton tunneling. Science 2006, 312, 237 to 241. Crossref. Nagel, ZD, Klinman, JP. Tunneling and dynamics in enzymatic hydride transfer. Chem. Reverend 2006, 106, 3,095 to 3,118. Crossreef. PubMed. 25. Olson, M.H., Parson, W.W. Warshell, A Dynamical Contributions to Enzyme Catalysis, Critical Tests of a Popular Hypothesis. Chem. Reverend 2006, 106, 1,737 to 1,756. Crossreef. PubMed. 26. Pu JZ, JO, JL. Trular DG multidimensional tunneling, recrossing, and the transmission coefficient for enzymatic reactions. Chem. Reverend 2006, 106, 3,140 to 3,169. Crossref. Hay S, Pudney, CR, Scruton, NS, Structural and Mechanistic Aspects of Flavor Proteins, Probes of Hydrogen Tunneling. Febs, J. 2009. 276th, 3930 to 3941. Cross-Ref. 28. Major DT, Haru A., Orville. AM, Valley, MP, Fitzpatrick PF. Gao JL differential quantum tunneling contributions in nitroalkane oxidase catalyzed and the uncatalyzed proton transfer reaction. Pros, Nuttall, Akad, CI, USA 2009, 106, 20,734 to 20,739. Cross-Ref. Camelin, J. Warshell, examining the case for the effect of barrier compression on tunneling, vibrationally enhanced catalysis, catalytic entropy and related issues. Febs Let, 2010, 584, 2,759 to 2,766. Cross-Ref. Camelin, SCL, Warshell, A. An analysis of all the relevant facts and arguments indicates that enzyme catalysis does not involve large contributions from nuclear tunneling. J. Phys. Org. Chem. 2010, 23, 677 to 684. Cross-Ref. 31. Smirnov I, Murok, LG, Nori, F. Kinetics of proton pumping in cytochrome C oxidase. J-Chem. Phys. 2009, 130, 06B620. Cross-Ref.
32. Smirnov I.
Savalev, Say. Nori F. Diffusion control generation of a proton motive force across a biomembrane. Phys. Reverend E. 2009, 80, 011,916. Cross-Reef. 33. Smirnov I. Nori F. Modeling the Q-cycle mechanism of transmembrane energy conversion. Phys. Biol. 2012-9, 016,011. Cross-Reef. Hay S. Johannesson, Lowe, Sutcliffe, M.J., Scruton, NS barrier compression and its contribution to both classical and quantum mechanical aspects of enzyme catalysis. Biophys, J. 2010, 98th, 121 to 128. Cross-Ref. PubMed. 35. Johannesson, Lowe, Hay S. Scruton, NS nuclear quantum tunneling in enzymatic reactions, an enzymologist's perspective. Phys. Chem. Chem. Phys. 2015, 17, 30,775 to 30,782. Cross-Reef. PubMed. 36. Johannesson, Lowe, Yorgu, AI, Scruton, NS, Hay, S. What are the signatures of tunneling in enzyme-catalyzed reactions? Faraday Discuss, 2019, 221, 367 to 378. Cross-Ref. PubMed. Quantum Rep. 2021, 3, 36, 37. Eyring H. Stern A. The application of the theory of absolute reaction rates to proteins. Chem. Reverend 1939, 24, 253 to 270. Cross-Ref. 38. Trular D.G. Garrett, B.C. Klippenstein S.J. Current status of transition state theory. J. Phys. Chem. 1996, 100, 12,771 to 12,800. Cross-Ref. Warshell A. Sharma P.K. Kato M. Shang Y. Liu H. Olson M.H. Electrostatic basis for enzyme catalysis. Chem. Reverend 2006, 106, 3,210 to 3,235. Cross-Ref. Gandor, R. Transition States of Biochemical Processes, Springer Science and Business Media, Berlin, Germany, 2013. Bell, R.P. The Tunnel Effect in Chemistry, Chapman and Hall, London, UK, 1980, pp 51 to 140. 42. Rickert, K.W., Klinman, J.P. Nature of Hydrogen Transfer in Soybean Lipoxygenase 1, Separation of Primary and Secondary Isotope Effects. Biochemistry 1999, 38, 12,218 to 12,228. Cross-Ref. Falder P.F., Tresodern G., Chauhan K.K., Scruton N., Sutcliffe, M.J., Hillier the Finch, Burton QM M&M studies show substantial tunneling for the hydrogen transfer reaction in methylamine dehydrogenase. J.M. Chem. Soc 2001, 123, 8604 to 8605. Cross Reef. 44. Trula D.G., Gao J.L., Garcia Viloca M., Alhambra C., Corchado J., Sanchez, M.L., Paulson T.D., Ensemble Averaged Variational Transition State Theory with Optimized Multidimensional Tunneling for Enzyme Kinetics and Other Condensed Phase Reactions. Int. J. Quant. Chem. 2004, 100, 1136 to 1152. Cross Reef. 45. Huang J.K. Warshell. A quantized classical path approach for calculations of quantum mechanical rate constants. J. Phys. Chem. 1993, 97, 10,053 to 10,058. Cross-ref. Mavri J. M.H. Warshell. A simulation of tunneling in enzyme catalysis by combining a biased propagation approach and the quantum classical path method, application to lipoxygenase. J. Phys. Chem. B. 2008, 112, 5,950 to 5,954. Cross Reef. PubMed. Garcia Viloca M. Gao J. Reaction path energetics and kinetics of the hydride transfer reaction, catalyzed by dihydrofolate reductase. Biochemistry 2003, 42, 13,558 to 13,575. Cross Reef. Pangjay Pujay, Gao J, Trula DG, Aliman RK hydride transfer reaction, catalyzed by hyperthermophilic dihydrofolate reductase, is dominated by quantum mechanical tunneling and is promoted by both inter- and intramonomeric correlated motions. J.M. Chem. Soc. 2006, 128, 8,015 to 8,023. Cross-ref. Alhambra C., Corchado J.C., Villa J., Trula, DG quantum mechanical dynamical effects in an enzyme-catalyzed proton transfer reaction. J.M. Chem. ASOC. 1999, 121, 2,253 to 2,258. Cross-ref. Tresodern G. McNamara J.P. Moore M. Wang H. Burton. N.A. Hillier. IH calculations of hydrogen tunneling and enzyme catalysis. A comparison of liver alcohol dehydrogenase, methylamine dehydrogenase, and soybean lipoxygenase. Chem. Phys. Let 2002. 358, 489 to 494. Cross-ref. 51. Garcia Viloca M. Alhambra C. Trula DG, Zhao J hydride transfer catalyzed by xyloisomerase, mechanism and quantum effects. J. Comput. Chem. 2003, 24, 177 to 190. Cross-ref. Dole KM, Bender, Barr, Finke, RG the first experimental test of the hypothesis that enzymes have evolved to enhance hydrogen tunneling. J. Am Chem. SOK. 2003, 125, 10,877 to 10,884. Cross-ref. Dol KM Finke. RG a compelling experimental test of the hypothesis that enzymes have evolved to enhance quantum mechanical tunneling in hydrogen transfer reactions. The beta-neopentyl cobalamin system combined with prior adicobalamin data. Inorg. Chem. 2003, 42, 4,849 to 4,856. Cross-reef. 54. Lushkov, V. Aquivist. J. Computer simulation of primary kinetic isotope effects in the proposed rate-limiting step of the glyoxylase I-catalyzed reaction. J. Biol. Chem. 2,275, 22,657 to 22,662. Cross-reef. 55. Williams IH quantum catalysis. A comment on tunneling contributions for catalyzed and uncatalyzed reactions. J. Phys. Org. Chem. 2010 23, 685 to 689. Cross-Reef, 56. Olson, Imham, Mavri, J. Warshell. A transition state theory can be used in studies of enzyme catalysis, lessons from simulations of tunneling and dynamical effects in lipoxygenase and other systems. Phil, Trans, Roy, Esok, B-Bile, SCI, 2006, 361, 1417 to 1432. Cross-Ref, PubMed, 57. Sikorsky Rs, Wang L, Markham, Carr, Rajagopalan PTR, Benkovich, SJ, Cohen. A tunneling and coupled motion in the Escherichia coli dehydrofolate reductase catalysis. J. Am Chem. Esok, 2004, 126, 4778 to 4779. Crossreef. Kim H.S. Damo S.M., Lee Sai, Wemmer D., Klinman J.P. Structure and hydride transfer mechanism of a moderate thermophilic dihydrofolate reductase from Bacillus styrothermophilus and comparison to its mesophilic and hyperthermophilic homologs. Biochemistry 2005, 44, 11428 to 11439. Cross-Ref, PubMed, Benner, Sir, Sassi, So, Gotcha, EA Molecular Paleoscience, Systems Biology from the Past. ADV, Enzymol, Relat, Areas, Mol Bile, 2007, 75-1. Hall BG Simple and Accurate Estimation of Ancestral Protein Sequences, Proconnatal, ACAD, AD. 5431 to 5436. Crossref, PubMed, 61. Perez Jimenez R, Ingles Prieto R, Zhao Zem, Sanchez Romero I, Allegra Cebalada J, Kazuri P, Garcia Menyas S, Kapok TJ, Tanokura M, Holmgren A, and others. Single-molecule paleoenzymology probes the chemistry of resurrected enzymes. Nat. Struct. Mol Bile. 2011-18, 592 to 596. Cross-Reef. 62, 63. Thornton J.W. Resurrecting Ancient Genes. Experimental Analysis of Extinct Molecules. Nat Rev. Genet. 2004, 5, 366 to 375. Cross-Reef. DeVault, Don, Parks, J.H., Chance, B. Electron Tunneling in Cytochromes. Nature 1967, 215, 642 to 644. Cross-Ref. Quantum Rep. 2021, 337, 64. Liu H. Warshell, A Origin of the Temperature Dependence of Isotope Effects in Enzymatic Reactions. The Case of Dihydrofolate Reductase. J. Phys. Chem. B. 2007, 111, 7852 to 7861. Cross-Reef. 65. Cohen, A. Dihydrofolate reductase as a model for studies of enzyme dynamics and catalysis. F1000 Research 2015, 4 F1000 Faculty, Reva 1464. Crossreef. 66. Hams, Schiffer, S. Hydrogen tunneling and protein motion in enzyme reactions. ACC, Shem. Riz. 2006, 39, 93 to 100. Crossreef. PubMed. 67. Klinman J.P. Cohen, A Hydrogen Tunneling Links Protein Dynamics to Enzyme Catalysis. Anu. Reverend Biochem. 2013, 82, 471 to 496. Cross-Ref. Cross-Ref. Klinman, J.P., Offenbacher, R. Understanding Biological Hydrogen Transfer Through the Lens of Temperature-Dependent Kinetic Isotope Effects. ACC, Chem. 2018, 51, 1966 to 1974. Crossref. Hay, Hothi P. Johannesson Low, Masgrau L. Pang J. Lays D. Sutcliffe M.J., Scruton N.S. Atomistic Insight into the Origin of the Temperature Dependence of Kinetic Isotope Effects and H. Tunneling in Enzyme Systems is revealed through combined experimental studies and biomolecular simulation. Biochem, S.O. Set, Trans, 2008, 36, 16-21. Cross, Ref. 70. Offenbacher R., Sharma A., Doan, P.E., Klinman J.P., Hoffman B.M., the soybean lipoxygenase, substrate complex, correlation between the properties of tunneling-ready states and indoor-detected structures of ground states. Biochemistry 2020, 59, 901-910. Cross. 71. Hu S. Sudakov Avi, Harm Schiffer S. Klinman J.P. Enhanced rigidification within a double mutant of soybean lipoxygenase provides experimental support for vibronically non-adiabatic proton-coupled electron transfer models. ACS Cattle, 2017. 7, 3569-3574. Crossreef. Kuznetsov M., Olstrup J. Proton and hydrogen atom tunneling in hydrolytic and redox enzyme catalysis. Can. J. Chem. 1999, 77, 1085-1096. Crossreef. K. Klinman. J.P. Temperature-dependent isotope effects in soybean lipoxygenase. 1. Correlating hydrogen tunneling with protein dynamics. J.M. Chem. ASOC. 2002, 124, 3865-3874. Crossreef. 7-4. Johannesson. Low. Iribo T. Stjoin. M. Johansson O. Hammerstrom L. The kinetic effect of internal hydrogen bonds on proton-coupled electron transfer from phenols. A theoretical analysis with modeling of experimental data. J. Phys. Chem. B. 2009, 113, 16,214-16,225. Cross-ref. 75. Hatcher E. Sudakov. A. Harms Schiffer S. Known adiabatic proton-coupled electron transfer reactions, impact of donor receptor vibrations, reorganization energies, and couplings on dynamics and rates. J. Phys. Chem. B. 2005, 109, 18,565-18,574. Cross-ref. 76. Meyer M.P., Tomczyk, Dr., Klinman, J.P. Enzyme structure and dynamics affect hydrogen tunneling, the impact of a remote side chain. I-553 in soybean lipoxygenase 1. Proconnatal ACAD, AD. USA 2008, 105, 1146-1151. Crossreef. PubMed 77 Pleasant, Pudney C.R., Johannesson. Low, Sutcliffe, M.J., Hay, S., Scruton, N.S., direct analysis of donor-acceptor distance and relationship to isotope effects, and the force constant for barrier compression in enzymatic H-tunneling reactions. J. M. Chem. ESOC, 2010, 132nd, 11,329 to 11,335. Crossref. 78. Hatcher E. Sudakov, A.V., Hams Schiffer, S. Proton-coupled electron transfer in soybean lipoxygenase, dynamical behavior and temperature dependence of kinetic isotope effects. J. M. Chem. SOCAR. 2007, 129, 187 to 196. Cross-ref. PubMed, Johannesson Low, Hay, S., Scruton, N.S., Sutcliffe M.J. Proton tunneling in aromatic amine dehydrogenase is driven by a short-range sub-picosecond promoting vibration, consistency of simulation and theory with experiment. J. Phys. Chem. B. 2007, 111, 2631 to 2638. Crossref. 80. N.S., Sutcliffe, M.J. How does pressure affect barrier compression and isotope effects in an enzymatic hydrogen tunneling reaction? Anju. Chem. Intid Engel. 2011, 50, 2129 to 2132. Crossref. 81. Hay S. Johannesson. Low, Hothy, P. Sutcliffe M.J. Scruton N.S. Pressure effects on enzyme-catalyzed quantum tunneling events arise from protein-specific structural and dynamic changes. J. M. Cross. SOC, 2012, 134, 9749 to 9754. Cross Reef, 82. Hay S. Pudney, CR. Sutcliffe, MJ. Scruton NS probing active site geometry using high pressure and secondary isotope effects in an enzyme-catalyzed, deep H-tunneling reaction. J. Phys. Org. Chem. 2010, 23, 696 to 701. Cross.
83.
Hay S. Scruton. NS incorporation of hydrostatic pressure into models of hydrogen tunneling highlights a role for pressure-modulated, promoting vibrations. Biochemistry, 2008, 47, 9880 to 9887. Cross-ref. 84, Hay S. Sutcliffe, MJ, Scruton, NS promoting motions in enzyme catalysis probed by pressure studies of kinetic isotope effects. Proconatal, ACAD, AD. USA 2007, 104, 507 to 512. Cross Reef. 85. Pudney CR, McGrory T, Lafitte P, Pang JY, Hay S, Lees D, Sutcliffe MJ. Scruton NS parallel pathways and free energy landscapes for enzymatic hydride transfer probed by hydrostatic pressure. Chem Biochem 2009-10, 1379 to 1384. Cross Reef. 87. E. Lee, C.C., eds. Elsevier, London, UK, 1984, Volume 6, pp. 67 to 105. Northrop, D.B. Unusual Origins of Isotope Effects in Enzyme-Catalyzed Reactions. Phil, Trans, R. Soc. B. 2006, 361, 1341 to 1349. Cross-ref. PubMed, Isaacs Enns, Javade, K. Ranelagh, E-Pressure Effects on Proton Tunneling. Nature 1977, 268, 372. Cross-Reef. PubMed. 89. Northrop. DB Effects of High Pressure on Isotope Effects and Hydrogen Tunneling. J. M. Chem. ESOC. 1999, 121, 3521 to 3524. Cross-Ref. 90. Hoven R. Hayes DJ. Hay S. Scruton NS. Does the pressure dependence of kinetic isotope effects report usefully on dynamics in enzyme H-transfer reactions? Febs J. 2015, 282, 3243 to 3255. Cross-ref. PubMed. Quantum Rep. 2021, 338. Ledwood D.A. Effects of Pressure on Protein Structure. High Press. Res. 2019, 391 to 400. Crossref. Ruiz Pernia J.J. Bihari E. Luke Lip. Loveridge. E.J. Tunon. I. Molina Fythe. Alaman RK Minimization of Dynamic Effects in the Evolution of Dihydrofolate Reductase. Chem. AD. 2016, 7. 3248 to 3255. Crossref. Warshell A. Bora. RP Perspective, Defining and Quantifying the Role of Dynamics in Enzyme Catalysis. J. Chem. Phys. 2016, 144, 180,901. Crossref. Warshell. A. Electrostatic origin of the catalytic power of enzymes and the role of pre-organized active sites. J. Biol. Chem. 1998, 273, 27,035 to 27,038. Crossreeve. 94. Schramm. V.L. Schwartz. S.D. Promoting Vibrations and the Function of Enzymes. Emerging Theoretical and Experimental Convergence. Biochemistry. 2018, 57, 3,299 to 3,308. Crossref. Johannesson, Lowe, Scruton, N.S., Sutcliffe, M.J. The enzyme aromatic amine dehydrogenase induces a substrate conformation crucial for promoting vibration that significantly reduces the effective potential energy barrier to proton transfer. J.R. Sock, Interface 2008, 5, Supple 3, S225-232. Crossref 96, Scruton Ns, Groot, ML, Highs, DJ excited state dynamics and catalytic mechanism of the light-driven enzyme protochlorophyllide oxidoreductase, Phys, Chem, Phys, March 14, 2012, 8818 to 8824. Crossref 97. Hunter, CN making light work of enzyme catalysis, protochlorophyllide oxidoreductase. Trends Biochem, AD, March 30, 2005, 642 to 649. Crossref, PubMed, 98. Menon B.R., Waltho, J.P., Scruton, N.S., Hayes, DJ cryogenic and laser photoexcitation studies identify multiple roles for active site residues in the light-driven enzyme protochlorophyllide oxidoreductase. J. Bile, Chem, 2009, 284, 18,160 to 18,166. Crossref, PubMed, Highs DJ, Sakuma, M, Devissa, SP, Scruton, NS nuclear quantum tunneling in the light-activated enzyme protochlorophyllide oxidoreductase. J. Bile, Chem, 2009, 284, 3,762 to 3,767. Crossref, PubMed, 101. Hayes, DJ, Sakuma, M, Scruton, NS solvent-slaved protein motions accompany proton, but not hydride tunneling, in light-activated protochlorophyllide oxidoreductase. Anju, Chem, Inted Engel, 2009, 48, 3,850 to 3,853. Crossref, 102. Hoven R. Hardman, S.J. Hayes, D.J., Scruton NS cross-species analysis of protein dynamics associated with hydride and proton transfer in the catalytic cycle of the light-driven enzyme protochlorophyllide oxidoreductase. Biochemistry 2016, 55, 903 to 913. Crossref, PubMed, Hayes D.J., Hardman S.J., Hedison T.M., Hoven R., Greetham G.M., Tari M., Scruton N.S. excited state charge separation in the photochemical mechanism of the light-driven enzyme protochlorophyllide oxidoreductase. Anju, Chem, Inted Engel, 2015, 54, 1,512 to 1,515. Crossref, 104. Menon B.R., Hardman, S.J., Scruton, N.S., Highs DJ multiple active site residues are important for photochemical efficiency in the light-activated enzyme protochlorophyllide oxidoreductase, PURE. J. Photochem, Photobile, B. 2016, 161, 236 to 243. Crossref, 105. Menon Bira, Davison, Pa, Hunter, CN, Scruton, NS, Hayes, DJ mutagenesis alters the catalytic mechanism of the light-driven enzyme protochlorophyllide oxidoreductase. J. Bile, Chem, 2010, 285, 2,113 to 2,119. Crossref, Hayes DJ, Levy, C, Sakuma, M, Robertson, DL, Scruton, NS, a twin-track approach, has optimized proton and hydride transfer by dynamically coupled tunneling during the evolution of protochlorophyllide oxidoreductase. J. Bile, Chem, 286, 11,849 to 11,854. Crossref, 106, 2011, 107. Hayes DJ, Heathcote P, Rigby, C. Palacios, Ma, Van Grondel, R. Hunter, CN. The first catalytic step of the light-driven enzyme protochlorophyllide oxidoreductase proceeds via a charge transfer complex. J. Bile, Chem, 2006, 281, 26,847 to 26,853. Crossref, PubMed, Garone A, Archipoa N, Zipfel, PF, Herman G, Dietzek B. Plant protochlorophyllide oxidoreductases A and B, catalytic efficiency and initial reaction steps. J. Bile, Chem, 2015, 290th, 28,530 to 28,539. Crossref, PubMed 109. Archipoa N. Cutter, R.J., Hayes, D.J., Scruton N.S. Stepwise hydride transfer in a biological system. Insights into the reaction mechanism of the light-dependent protochlorophyllide oxidoreductase. Anju. Chem. 2018, 130, 2712 to 2716. Cross-ref. 110. Zhang S., Hayes D.J., Feng L., Sun W., Johannesson Lo, Liu H., Levy C.W., Li X., Yang J. UX, and others, structural basis for enzymatic photocatalysis in chlorophyll biosynthesis. Nature 2019, 574, 722 to 725. Cross-ref. PubMed 111. Dong C's, Zhang WL, Wang Q, Li E's, Wang X, Zhang M, Liu L crystal structures of cyanobacterial light-dependent protochlorophyllide oxidoreductase. Proconnatal ACAD, AD. USA 2020, 117, 8,455 to 8,461. Cross-ref. PubMed 112. Basham G.A., Calvin M., The Path of Carbon in Photosynthesis. In DiCO2 assimilation, the assimilation of carbon dioxide. Springer, Berlin Heidelberg, Germany, 1960. pp 884 to 922. 113. Bolton, J.R., Hall, Do the Maximum Efficiency of Photosynthesis. Photochem, Photobiol, 1991, 53, 545 to 548. Cross-Re, 114. Zhu, X.G. Long, S.P. Ort, Dr. What is the maximum efficiency with which photosynthesis can convert solar energy into biomass? Kerr, Open, Biotechnol, 2008, 19, 153 to 159. Cross-Reef, 115. Chen, G.Y., Lambert, N., Lee, C.M., Chen, Y.N., Nori F. Rerouting excitation transfers in the Fenner-Matthews-Olson complex. Phys. Reverend E. 2013. 88. Vorand E. 2000, 120. Cross-Reef, 116. Panichayankun G. Voronine D.V. Abramovicius D. Karam J.R. Lewis N.H. Mukamel S. Engel-Gius Direct Evidence of Quantum Transport in Photosynthetic Light Harvesting Complexes. Proconnatal ACAD, AD. USA 2011, 108, 20,908 to 20,912. Cross-Ref, 117. Forster, T-transfer mechanisms of electronic excitation energy, Radiat, Res, Supple, 1960, 326 to 339. Saint-Dixhuy, Perrin, F. Théorie quantique des transferts d'activation entre molécules de même espèce. Cas de solution fluorescente. Anne Phys. Dix Neuf Cent Trent 2. Dix 283 Quantum Rep. 3, 39. 119. Forster T. Energiewanderung und Fluoreszenz. Naturwissenschaften 1946. 33. 166 to 175. Cross-Ref. 120. Alden R. Johnson E. Nagarajan Faith. Parson W. Law C. Cogdell R. Calculations of spectroscopic properties of the LH2 bacteriochlorophyll protein antenna complex from Rhodopseudomonas acidophila. J. Phys. Chem. B. 1997. 101. 4,667 to 4,680. Cross-Riff. Chachis-Willis M. Kuhn. O. Pulleritz T. Sundstrom Vive excitans in photosynthetic purple bacteria, wave-like motion or incoherent hopping. J. Phys. Chem. B. 1997. 101, 7,275 to 7,283. Cross-Reef. 122. Zhang S. Newton, M.D. Silby, R.J. Multichromophoric Forster resonance energy transfer from B800 to B850 in the light harvesting complex 2. Evidence for subtle energetic optimization by purple bacteria. J. Phys. Chem. B 2007. 111, 6807 to 6814. Cross-refer. PubMed. Ulbricht C, Janssen TL, Liebers J, Attar M, Strumpfer J, Schulten K, Nestor J, Kleinekathofer U, from atomistic modeling to excitation transfer and two-dimensional spectra of the FMO light harvesting complex. J. Phys. Chem. B 2011, 115, 8609 to 8621. Cross-ref. PubMed. 124, Cogdell RJ, Prince SM, Freer A, Isaacs, NW, Shear H structure-based calculations of the optical spectra of the LH2 bacteriochlorophyll protein complex from Rhodopseudomonas acidophila. Photochem. Photobile. 1996, 64, 564 to 576. Cross-ref. 125. Scholz GD, Gould Iyer, Cogdell, RJ, Fleming Ger ab initio molecular orbital calculations of electronic couplings in the LH2 bacterial light harvesting complex of RPS. Acidophila. J. Phys. Chem. B. 1999. 103, 2543 to 2553. Cross-ref. Fleming. G.R. Van Grondel, R. Femtosecond spectroscopy of photosynthetic light harvesting systems. Kerr. Open. Struct. Bile. 1997, 738 to 748. Cross-reef. 128. Van Amerongen H. Van Grondel, R. Photosynthetic excitons. World Scientific. Singapore. 2000. 129. Nakamura, E. Aratani, N. Osuka, A. Cyclic porphyrin arrays as artificial photosynthetic antenna. Synthesis and excitation energy transfer. Chem. Soc. Reverend 2007. 36, 831 to 845. Cross, Reef. 130. Facioli F. Dinshaw, R. Arpin, PC. Scholes GD photosynthetic light harvesting, excitance and coherence. J.R. Soc. Interface 2014, 11, 20,130,901. Cross Reef. Champa C. Rafiq, Scholz GD from coherent to vibronic light harvesting in photosynthesis. Kerr. Open. Chem. Bile. 2018, 47, 39 to 46. Cross-ref. PubMed. 132. Has U. CP. The electronic couplings in electron transfer and excitation energy transfer. ACC. Chem. Res. 2009. 42, 509 to 518. Cross-reef. PubMed. 133rd. Sate J. Manfcal. T. Ultrafast energy transfer with competing channels, non-equilibrium Forster and modified Redfield theories. J. Chem. Phys. 2017. 146. 174,109. Cross-reef. PubMed. 134th. Tao, MJ, Zhang, NN, Wen, PY, Dung, FG, AI, Q, Long, GL coherent and incoherent theories for photosynthetic energy transfer, SEI, Bull, 2020, 65th, 318 to 328. Crossref. 135. Ishizaki A. Tanemura. Why quantum dynamics of systems strongly coupled to low-temperature colored noise bath, reduced hierarchy equations approach. J. Phys. ESOC. JPN. 2005, 74, 3131 to 3134. Crossreef. 136. Ishizaki A. Fleming. GR theoretical examination of quantum coherence in a photosynthetic system at physiological temperature. Proconatal ACAD. AD. USA 2009. 106. 17,255 to 17,260. Crossreef. 137. Lambert N. Ahmed S. Sirio. M. Nori F. Modeling the ultra-strongly coupled spin boson model with unphysical modes. Nat. Common. 2019. 10. 1-9. Cross-Ref. 138 Chin, or Huelga, SF Plenio, MB Efficient Simulation of Strong System-Environment Interactions. Phys. Reverend Let. 2010. 105. 050,404. Cross-Reef. 139. Chin, or Rivas. A. Huelga, SF Plenio. MB exact mapping between system reservoir quantum models and semi-infinite discrete chains using orthogonal polynomials. J. Math. Phys. 2010. 51. 092,109. Cross-Ref. 140. Chin or Huelga, SF Plenio, MB chain representations of open quantum systems and their numerical simulation with time-adaptive density matrix renormalization group methods. In Semiconductors and Semimetals, Elsevier, Amsterdam, the Netherlands, 2011, Volume 85, pp. 115 to 143, 141, Chin A, Pryor J, Rosenbach R, Caicedo Solar F, Huelga SF, Plenio MB. The role of non-equilibrium vibrational structures in electronic coherence and recoherence in pigment protein complexes. Nat. Phys. 2013, 9. 113 to 118. Cross-Reef. 142. Gelzinis A, Orgulis R, Butkus, Feeve, Robert B, Valkunus, L. Two-dimensional spectroscopy for non-specialists. Biochim. Biophys, Acta Bionerg. 2019, 1860, 271 to 285. Cross-Ref. PubMed. 143. Alodi, Ma. Engel GS quantum coherences reveal excited state dynamics in biophysical systems. Nat. Reverend Chem. 2019. 3. 477 to 490. 144. Lewis, K.L., Ogilvy, J.P. Probing photosynthetic energy and charge transfer with two-dimensional electronic spectroscopy. J. Phys. Chem. Let. 2012. 3. 503 to 510. Cross-Ref. Pub. Med. 145. Ostromov E. Mulvaney, R.M., Cogdell, R.J., Scholes GD broadband 2D electronic spectroscopy reveals a carotenoid dark state in purple bacteria. Science 2013, 340, 52 to 56. Cross-Ref. 146. Chin or Huelga, SF Plenio, MB quantum metrology in non-Markovian environments. Phys. Reverend Let. 2012, 109, 233,601. Quantum Rep. 2021, 340, 147. Bagbanzadeh S. Kassal, distinguishing the roles of energy funneling and delocalization in photosynthetic light harvesting. Phys. Chem. Phys. March 18, 2016, 7,459 to 7,467. Cross-Reef. 148. Engel Gs. Calhoun TR. Red L. Auntie K. Manvcal T. Cheng Y. C. Blankenship. Re. Fleming GR evidence for wave-like energy transfer through quantum coherence in photosynthetic systems. Nature, 2007, 446, 782 to 786. Cross-Reef. 149. Sarovar, M. Ishizaki, A. Fleming GR. Whaley KB quantum entanglement in photosynthetic light harvesting complexes. Nat. Phys. 2010, 6. 462 to 467. Cross-Reef. 150. Sao J, Cogdell RJ, Coca DF, Duan HG, Howard J, Kleinekathofer U, Janssen, TL, Manvcal T, Miller R&D, Ogilvy JP Quantum Biology Revisited. ADV 2026, EAAs 4888. Cross-Ref. 151. Li H, Cheng, YC, Fleming GR coherence dynamics in photosynthesis, protein protection of excitonic coherence. Science 2007, 316, 1462 to 1465. Cross, Reef. 152. Ryu is, Dong H. Fleming Gr. Role of electronic vibrational mixing in enhancing vibrational coherences in the ground electronic states of photosynthetic bacterial reaction center. J. Phys. Chem. B. 2014, 118, 1381 to 1388. Cross, Reef. Pub. 153. Ma F. Romero E. Jones Mr. Novodereshkin VI. Van Grondel R. Both electronic and vibrational coherences are involved in primary electron transfer in bacterial reaction center. Nat. Common. 2019. 10, 1-9. Crossref. PubMed. 154. Christensen N. Kaufman, H.F. Pulleritz T. Mankell T. Origin of long-lived coherences in light harvesting complexes. J. Phys. Chem. B. 2012, 116, 7449 to 7454. Crossref. 155. Thihug E. Tempelar R. Alcazar MJ. Zidek K. Bina D. Nestor J. Janssen TL. Zygmantas D. Identification and characterization of diverse coherences in the Fenner-Matthews-Olson complex. Nat. Chem. 2018. 10-780 to 786. Crossref. 156. Dean J.C., Mirkovich, T. Toa, Zess, Oblinsky D.G., Scholes G.D. Vibronic enhancement of algae light harvesting. Chem 2016, 1, 858 to 872. Crossref. 157. Tiwari Fifth, Peters, W.K., Jonas, DM electronic resonance with anti-correlated pigment vibrations drives photosynthetic energy transfer outside the adiabatic framework. Proconnatal ACAD, AD. USA 2013, 110th, 1200, and 3 to 1208. Cross Reef, 158. Palevchek D., Edland P., Westenhoff S., Zygmantas D. Quantum coherence as a witness of vibronically hot energy transfer in bacterial reaction center. ADV 2017. 3. E1603141. Cross Ref. 159. Calhoun T.R., Ginsberg N's, Schlau Cohen G's, Chang I.C., Balatari M., Bassi R., Fleming Gr-quantum coherence enabled determination of the energy landscape in light harvesting complex. 2nd. J. Phys. Chem. B. 2009. 113, 16,291 to 16,295. Cross-ref. 160. Kalini E., Wong Sai, Wilk K., Kermi PM, Bruma P., Scholes GD coherently wired light harvesting in photosynthetic marine algae at ambient temperature. Nature, 2010, 463, 644 to 647. Cross Reef. 161. Romero E., Ogulis R., Novodereshkin 6, Ferretti M., Team J, Zygmantas D, Van Grondel R. Quantum coherence in photosynthesis for efficient solar energy conversion. Nat. Phys. 2014, 10, 676 to 682. Cross Reef. 162. Panetchayankun G, Hayes D, Franstad Carr, Karam Jr., Harel E. Wen J, Blankenship Re, Engel G's long-lived quantum coherence in photosynthetic complexes at physiological temperature. Proconnatal ACAD, AD. USA 2010, 107th, 12,766 to 12,770. Cross-ref. PubMed. And 63. Chen J, Sakurai A, Shiq, Gardner at, Kuhn O, Cogdell RJ, Pulleritz T before Forster. Initial excitation in photosynthetic light harvesting. Chem. C. 2019-10, 7,923 to 7,928. Crossref. Crossreef. Urgenjuro S, Gurongan K, SAR, RG, Blankenship Re, Harel E. Electronic Coherence Lifetimes of the Fenner-Matthews-Olson Complex and Light Harvesting Complex Second Chem 80 2019 10,503 to 10,509 Cross-Ref PubMed 165 Hildner R, Brinks D, Nida, JB, Cogdell RJ, Van Hulst, NF Quantum Coherent Energy Transfer over Varying Pathways in Single Light Harvesting Complexes. Science 2013, 340, 1448 to 1451. Cross-Ref. Cross-Ref, Chin A, Huelga, SF, Plenio, MB Coherence and Decoherence in Biological Systems, Principles of Noise-Assisted Transport and the Origin of Long-Lived Coherences. Phylos, Trans, RSOC, Amath, Phys, Eng Psi. 2012, 370, 3638 to 3657. Crossref. PubMed, 167. Harel E. Engel, GS Quantum Coherence Spectroscopy reveals complex dynamics in bacterial light harvesting complex 2, LH2. Proconatal ACAD, AD. USA, 2012. 109, 706 to 711. Crossreef. 168. Fisher M. Gutierrez Medina, B. Risen M. Observation of the quantum xeno and anti-xeno effects in an unstable system. Phys. Reverend Lett. 2001, 87, 040,402. Crossref. 169. Rebentrost P. Moseni M. Casal, I. Lloyd S. Asperu Guzik A. Environment-Assisted Quantum Transport. New J. Phys. 2009, 11, Seal 33003. Cross-Ref. 170. Moseni M. Shabani A. Lloyd S. Rabbits H. Energy Scales Convergence for Optimal and Robust Quantum Transport in Photosynthetic Complexes. JChem. Phys. 2014, 140, 01, B609, 601. Cross-Ref. 171. Jang S.J. Robust and Fragile Quantum Effects in the Transfer Kinetics of Delocalized Excitons between B850 units of LH2 complexes. J-Phys. Chem. Let. 2018-9, 6,576 to 6,583. Crossref. PubMed. 172. Prokhorenko, Zix, Nagy M, Washchuk Se, Brown, Elez, Burge R.R., Miller R&D Coherent Control of Retinal Isomerization in Bacteria Hodopsin. Science 2006, 313, 1,257 to 1,261. Cross-Reef. Science 273. W. Dane, M. Conversion of Energy in Halobacteria, ATP Synthesis and Phototaxis. Arch. Microbial. 1996, 166, 1-11. Cross-Ref. PubMed. 174. Orin A. The Order Halobacterialis. Prokaryotes 2006, 3, 113 to 164. 175. Friedrich M., Silfston P., Parkinen S., Parkinen J., Jaskalainen T. Color-sensitive retina based on bacteria Hodopsin. Biosystems 2054, 131 to 140. Crossref. Quantum Rep. 2021, 341, 176. HAMP. N. Bacteriohodopsin as a photochromic retinal protein for optical memories. Chem. Reverend 2000, 100th, 1755 to 1776. Crossref. PubMed. 177. Margesin R. Shinner F. Potential of halotolerant and halophilic microorganisms for biotechnology. Extremophiles 2001, 573 to 83. Crossref. 178. Rakovich R. Sukhanova A. Bouchonville N. Lukashev E. Oleinikov, Feith, Artemiev M. Lesniak Feith, Gaponik N. Molinari M. Troyon M resonance energy transfer improves the biological function of bacteriohodopsin within a hybrid material built from purple membranes and semiconductor quantum dots. NanoLet 2010-10, 2640 to 2648. Crossref 179. Ghosh PK, Smirnov, I, Nori F, modeling light-driven proton pumps in artificial photosynthetic reaction centers. JChem, Phys, 2009, 131, 07B610. Crossref 180. Prope A, Li YC, Asperu Guzik A, Berlinget CP, Chang CJ, Cogdel R, Doyle Ag, Flick J, Gabor NM, Van Grondel are bioinspiration in light harvesting and catalysis. Nat, Reverend Mater, 2020, 5, 828 to 846. Crossref. Leash, Matula AJ, Hu G, Troiano JL, Karpovich CJ, Crabtree, RH, Batista versus Brudvig GW strongly coupled phenazine porphyrin dyads, light harvesting molecular assemblies with broad absorption coverage. ACS Apple, Mater. Interfaces 2019, 11, 8000 to 8008. Crossref. Prinz J Apple, 8000, M Kellerby, Sen, M, Held M, Kudera, JD, Schutzi Noe, F Markov models of molecular kinetics, generation and validation. JChem. Phys. 2011. 134, 174,105. Crossref. PubMed 183. Ghosh PK, Smirnov, I, Nori. F quantum effects in energy and charge transfer in an artificial photosynthetic complex. JChem. Phys. 2011. 134, 06, B611. Crossref. PubMed on 84, Ghosh, S, Elefountain. Am, Frank, Ha. Beck WF structural tuning of quantum decoherence and coherent energy transfer in photosynthetic light harvesting. JPhys. Chem. Let. 2018, 9, 5071 to 5077. Crossref. 185. Delor M, Di J, Roberts TD, Rogers JR, Hamed SNM. Neaton JB, Geisler, PL, Francis, MB, Ginsberg Ns, exploiting chromophore protein interactions through linker engineering to tune photo-induced dynamics in a biomimetic light harvesting platform. J. M. Chem. SOC, 2018, 140, 6278 to 6287. Crossref. 186. Wang L, Griffin GB, Zhang A, Zhayev, Williams Nair, Jordan RF, Engel G's controlling quantum beating signals in 2D electronic spectra by packing synthetic heterodimers on single-walled carbon nanotubes. Nat. Chem. 2017, 9, 219. Crossref. 187. Freicius V, Tretiak S, Makov, DV, Shalashalin DV, Fernandez Alberti S vibronic quantum beating between electronic excited states in a heterodimer. JPhys. Chem. B 2020. 124, 3992 to 4001. Crossref. 188. Mac Cleese, C. UZ, Ezamoto NN, Kolodziash C. Meti B. Bandari S. Dunyats, BD Burda C. Pashik M. Excitonic interactions in bacteriochlorine homodyads enable charge transfer, a new approach to the artificial photosynthetic special pair. JPhys. Chem. B 2018, 122, 4131 to 4140. Crossref. 189. Tiwari V, Matutsia, Kanara, UZ, Peteshek M, Boshan DF, Holton D, Kamaya C, Ogilvy JP strongly coupled bacteriochlorine dyad studied using phase-modulated fluorescence-detected two-dimensional electronic spectroscopy. Opt. Express 2018 26 22327 to 22341. Crossref. 190. Shoji S, Tamiyaki, H. Supramolecular light harvesting antenna system by co-aggregates of zinc, bacterio, chlorophyll derivatives with biomimetic chlorosomal self-assemblies. Dye. Pigment. 2019, 160, 514 to 518. Cross-ref. 191. Shoji S. Nomura. Y. Tamiaki. H. Heterodimers of zinc and free base chlorophyll derivatives co-assembled in biomimetic chlorosomal J aggregates. Photochem. Photobiol. Sci. 2019, 18. 555 to 562. Cross-ref. PubMed. Pandya R. Chen Rai. Keminal A. Thomas T. Thampi A. Tano A. Richter J. Shivana R. Deschler F. Schneiderman C. Observation of vibronic coupling-mediated energy transfer in light-harvesting nanotubes, stabilized in a solid-state matrix. J. Phys. Chem. Let. 2018. 9, 5604 to 5611. Cross-ref. PubMed 192, 193. Kim Taham S. Leash Hong Y. Kim D. Enhancement of exciton transport in porphyrin aggregate nanostructures by controlling the hierarchical self-assembly. Nanoscale 2018, 10, 16,438 to 16,446. Cross-ref. 194. Lloyd S. Moseni M. Symmetry-enhanced supertransfer of delocalized quantum states. New J. Phys. 2010, 12, 075,020. 195th. Chuang C. Li C. Moy J. M. Nesta J. Sao J. Quantum diffusion on molecular tubes, universal scaling of the 1D to 2D transition. Phys. Reverend Let. 2016, 116, 196,803. Cross-ref. PubMed. 196. Lim J. Palevchek D. Caicedo Solar F. Lincoln CN. Pryor J. Von Belepsch H. Huelga SF. Plenio MB. Zygmontas D. Howard J. Vibronic origin of long-lived coherence in an artificial molecular light harvester. Nat. Commune. 2015. 6, 1-7. Cross-ref. PubMed. 197. Butkus Vith. Alster J. Basinskate. E. Augulus RN. Newhouse P. Valkunas L. Anderson. HL. Abramavicius D. Zygmantas D. Discrimination of diverse coherences allows identification of electronic transitions of a molecular nanoring. J. Phys. Chem. Let. 2017. 8, 2,344 to 2,349. Cross-ref. Cheng, I.C. Fleming, GR Dynamics of Light Harvesting in Photosynthesis. Anu, Reverend Phys, Chem, 2009, 60, 241 to 262. Cross-ref. Calhoun, T.R. Fleming, GR Quantum Coherence in Photosynthetic Complexes. Phys. Status Solidi, B. 2011, 248, 199. 833 to 838. Cross-ref. 200. Van Grondel, R. Novodereshkin, Vi Quantum Effects in Photosynthesis. Procedia Chem, 2011, 3, 198 to 210. Cross-ref. 201. Lambrev Ph. Akhtar P. Tan, HS Insights into the mechanisms and dynamics of energy transfer in plant complexes from two-dimensional electronic spectroscopy. Biochim. Biophys. Acta Bionog, 2020, 1861, 148,050. Light Harvesting, Cross-ref, PubMed, 202. Chen, G.Y. Lambert, N. Shear, Y.A. Liu, M. H. Chen, Y.N. Nori F. Plasmonic Biosensing for the Fenner-Matthews-Olson Complex. AD. Rep. 2017, 7, 39,720. Cross-ref. PubMed. Quantum Rep. 2021, 342, 203. Li, C.M. Lambert, N. Chen, I.N. Chen, G.Y. Nori F. Witnessing Quantum Coherence from Solid State to Biological Systems. AD. Rep. 2012, 2, 885. Crossref. 204. Yuanzhou J. Critch, J.J., Aspuru Guzik, AA witness for coherent electronic versus vibronic-only oscillations in ultra-fast spectroscopy. JChem. Phys. 2012. 136, 234,501. Crossref. PubMed. 205. Johnson as Yuanzhou J. Aspuru Guzik, A. Critch, J.J. Practical witness for electronic coherences. JChem. Phys. 2014. 141, 244,109. Crossref. 206. Hallowell B. Gutteridge, J.M. Free Radicals in Biology and Medicine, Oxford University Press, New York, NY, USA, 2015. 207. Cannon K. Jane, SK Oxidative Stress and Apoptosis, Pathophysiology, 2007, 153 to 163. Crossref. Wright S.W., Kim H.P., Hotzel A., Park J.W., Nakahira K., Wang X. Choi and Mechanisms of Cell Death in Oxidative Stress. Antioxid. Redox Signal. 2007 9, 49 to 89. Crossref. 209. Chen Wei, Zhou Zi, Min W. Mitochondria, Oxidative Stress and Innate Immunity. Front. Physiol. 2018 9, 1487. 211. Suzuki, YJ, Foreman, HJ, Savanian are oxidants as stimulators of signal transduction. Free radic. Bile. Med. T97 22, 269 to 285. Cross Reef. 212. Buckle, W. Golding, BT radical enzymes. Incycle, Radic, Chem, Bile, Meta 2012. Crossref. 213. Conrad Kays, Manahan, CC, Crane, BR Photochemistry of Flavor Protein Light Sensors. Nat, Chem. Bile 2014, 10, 800 and 1 to 809. Crossreef. PubMed. 214. UE, Ulrich. T magnetic field effects in chemical kinetics and related phenomena. Chem. Reverend 1989, 89, 51 to 147. Crossref. 215. Hoare, P.J., Muritzen. H. The radical pair mechanism of magnetoreception. Anu, Reverend Biofees. 2016, 45, 299 to 344. Crossref. PubMed. Steiner, 216. Luxon. Enin, Ivanov, K.L., Sadovsky, V.M., Sagiv, RZ magnetic field effect on recombination of radicals diffusing on a two-dimensional plane. JChem. Phys. 2020, 152, 034,103. Crossref. 217. Sampson C., Keynes, R.H. Katnig, Doctor on the magnetosensitivity of lipid peroxidation, 2 versus 3 radical dynamics. Phys. Chem. Phys. March 21st, 2019, 13,526 to 13,538. Crossreef. 218% Brocklehurst B. McLaughlin. Ka free radical mechanism for the effects of environmental electromagnetic fields on biological systems. Int J. Radiat. Bile. 1996, 69, 3 to 24. Crossreef. 219. Jones R. Magnetic field effects in proteins. Mol. Phys. 2016 114, 1691 to 1702. Crossref. 220. Godbane S. Labib. A. Sackley M. Abdelmalek H. Bioeffects of static magnetic fields, oxidative stress, genotoxic effects, and cancer studies. Biomade Res. Int 2013, 600 and 2,987. Crossref. 221. Matson. M.O. Simcoe, M. Grouping of experimental conditions as an approach to evaluate effects of extremely low-frequency magnetic fields on oxidative response in vitro studies. Front. Public Health, 2014, 2, 132. Crossref. 222. Grissom, C.B. Magnetic Field Effects in Biology. A survey of possible mechanisms with emphasis on radical pair recombination. Chem. Reverend 1995, 95, 3-24. Cross-ref. 223. Muheim R., Bostrom J., Ackerson S., Liedvogel M. Sensory Mechanisms of Animal Orientation and Navigation. In Animal Movement Across Scales. Oxford University Press. Oxford, UK 2014, pp 179 to 194. 224. Fay, T.P., Lindoy, L.P., Manolopoulos, D. Hoare P. How Quantum is Radical Pair Magnetoreception.
Faraday Discuss. 2019, 221, 77-91.
Cross-ref. PubMed. 225. Woodward, J. Radical Pairs in Solution. Prog. React. Keenet. Mech. 2002. 27. 165-207. Crossref. 226. Rogers, CT Magnetic Field Effects in Chemical Systems. Pure Apple. Chem. 2009. 81. 19-43. Crossref. 227. Maeda K. Henbest KB. Sintelisi F. Kuprov I. Rogers CT. Lidlpa. Gusti. Timmel CR. Hoare PJ Chemical Compass Model of Avian Magnetoreception. Nature 2008. 453, 387-390. Crossreef. 228. Rogers, CT, Hoare, PJ Chemical Magnetoreception in Birds, the Radical Pair Mechanism. Proc, Natal, Uckard. CI, USA 2009, 106, 353-360. 229. Anakiva, P. Jasanoff, A. Magnetogenetics, Problems on the Back of an Envelope. Life 2016, 5, in 19,569. Cross Reef, 230. Meister, M. Physical Limits to Magnetogenetics. Life 2016, 5, 17,210. Cross. Life, Henbest, KBA Study of Spin Chemistry in Weak Magnetic Fields. Philos, Trans, R.Soc. Lund, Sir, Amath, Phys. Engsai, 2004. 362, 2,573-2,589. Cross Reef, 232. Biskup T. Schleicher E. Okafuji A. Link G. Hitomi K. Getzoff Ed. Weber S. Direct observation of a photo-induced radical pair in a cryptochrome blue light photoreceptor. Anju. Chem. Int Ed. 2009. 48. 404-407. Cross-ref. 233. Wilczko W. Munro. U. Ford H. Wilczko R. Red light disrupts magnetic orientation of migratory birds. Nature. 1993. 364. 525-527. Cross, Reef, 234. Phillips J. Said. O. Wavelength-dependent effects of light on magnetic compass orientation in Drosophila melanogaster. J. Comp. Physiol. A 1993, 172, 303-308. Cross Reef, PubMed, 235. Cutter R.J. Magul. K. Kenzie, U. Dick. B. A search for radical intermediates in the photocycle of love domains. Photochem. Photobile. SI 2015-14. 288-299. Crossref. PubMed. 236. Weber S. Light-driven enzymatic catalysis of DNA repair. A review of recent biophysical studies on photoliase. Biochim. Biophys. Acta Bionerg. 2005, 1707, 1-23. Crossref. PubMed. 237. Hay S. Brenner S. Cara B. Quinn Am. Rigby Say. Scruton NS Nature of the Energy Landscape for Gated Electron Transfer in a Dynamic Redox Protein. J. M. Chem. SOC. 2010. 132, 9,738 to 9,745. PubMed. Quantum Rep. 2021, 343. 238. Silverman, R&B Radical Ideas About Monoamine Oxidase, ACC, Chem. Res. March 28, 1995. 335 to 342. Cross-Ref. 239. Brown KL Chemistry and Enzymology of Vitamin B12. Chem. Reverend 2005, 105, 2075 to 2150. Cross-Ref. PubMed. 240. Marsh Eng, Patterson, MDP, Lee L-adenosyl radical, reagent and catalyst in enzyme reactions. Chem Biocheme Ayur. J. Chem. Bile. 2010, 11, 604. Cross-Reef. 242. Jones R. Woodward, J. R. Scruton, NS Continuous Wave, Photolysis Magnetic Field Effect Investigations with Free and Protein-Bound Alkylcobalamins. J. Am. Chem. Soc. 2009, 131, 17,246 to 17,253. Cross-Ref. Lukinovic V. Woodward J. R. Marafa, T.C., Shanmugam, M. Hayes, D.J., Hardman, S.J., Scruton N's, Hay S., Fielding A.J. Strones, A.R. Photochemical Spin Dynamics of the Vitamin B12 Derivative, Methylcobalamin. J. Phys. Chem. B. 2019. 123, 4,663 to 4,672. Cross-Reef. Pub. Kiprianov Jr. A. Doktorov A. Pertov P. Magnetic field effects on bistability and bifurcation phenomena in lipid peroxidation. Bioelectromagnetics 2015, 36, 485 to 493. Cross-Reef. PubMed. 245. Bujachenko Al. Kuznetsov Da. Shishkov RV spin biochemistry, magnetic isotope effect in the reaction of creatine kinase with CH3HGCl, J. Phys. Chem. A 2004, 108, 707 to 710. Cross-Reef. 246. Bujachenko Al. Kuznetsov, D.A., Orlova Markarian A.A. magnetic isotope effect of magnesium in phosphoglycerate kinase phosphorylation. Proconatal Acad. A.D. USA 2005, 102nd, 10,793 to 10,796. Cross-Ref. 247. Bujachenko Al. Kuznetsov, Dar, Breslavskaya, N.N., Orlova magnesium isotope effects in enzymatic phosphorylation. J. Phys. Chem. B. 2008, 112, 2,548 to 2,556. Cross-Ref. 248. Bujachenko Al. Kuznetsov the magnetic field affects enzymatic ATP synthesis. J. Am. Chem. ESOC. 2008, 130, 12,868 to 12,869. 249th. Crotty D. Silkstone G. Podar S. Ranson R. Prinamello A. Wilson M.T. Coe J. Re-examination of magnetic isotope and field effects on adenosine triphosphate production by creatine kinase. Proconatal Acad. A.D. USA 2012, 109th, 1,437 to 1,442. Cross-Reef. 250. Uselman R.J. Chavariaga C. Costello P.R. Procopio M. Ritz T. Dratz E.A. Single D.J. Martino C.F. The quantum biology of reactive oxygen species partitioning impacts cellular bioenergetics. A.D. Rep. 2016. 6. 38,543. Cross-Ref. 251. Uselman R.J. Hill I. Single D.J. Martino C.F. Spin biochemistry modulates reactive oxygen species, ROS, production by radio frequency magnetic fields. Pilos 1 2014, 9 in 93,065. Cross-Reef. Dodson C.A., Maeda K., Biscup T., Wedge C. Timmel CR magnetic field effects in flavor proteins and related systems. Interface focus 2013-3, 20,130,037. Cross reef 253. Evans U. Katnig Dr. Henbest, KB, Hoare, P. Mackenzie Sr. Timmel CR sub-millitesla magnetic field effects on the recombination reaction of flavin and ascorbic acid radicals. JChem. Phys. 2016. 145. 85,101. Cross Ref. PubMed. 255. Jones A.R. Hardman S.J. Hay S. Scruton Ns. Is there a dynamic protein contribution to the substrate trigger in coenzyme B12-dependent ethanolamine ammonia lyase? Anju. Chem. Inted. 2011. 50. 10,843 to 10,846. Crossref. Hughes Jar. Hardman S.J. Scruton N. Graham D.M. Woodward J.R. Jones. Our observation of the G mechanism resulting from the ultra-fast spin dynamics that follow the photolysis of coenzyme B12. JChem. Phys. 2019. 151. 201,102. Cross-ref. PubMed. Musewald C. Gilch P. Hartwich G. Pollinger Dammer F. Shear H. Michel BL Me. Magnetic field dependence of ultra-fast intersystem crossing, a triplet mechanism on the picosecond timescale. J.M. Chem. Soc. 1999. 121. 8,876 to 8,881. Cross, Reef. Mayeda K. Robinson A.J. Henbest K.B. Hogben H.J. Biskup T. Ahmad M. Schleicher E. Weber S. Timmel C.R. Hoare P.J. Magnetically sensitive light-induced reactions in cryptochrome are consistent with its proposed role as a magnetoreceptor. Proconnatal Acad. AD. USA 2012. 109. 4,774 to 4,779. Crossref. 258, 259. Shepard D.M. Lee J. Henbest K.B. Neil Sr. Maida K. Story J. Schleicher E. Biskup T. Rodriguez R. Weber S. millitesla magnetic field effects on the photocycle of an animal cryptochrome. AD. Rep 2017-7. 42,228. Cross Reef. 260. Sherard R. M. Morellini N. Jordan N. L. Esawi M. Arthort L. D. Niesner C. Royer F. Klarsfeld A. Dulazmi M. Vichak J. Low-intensity electromagnetic fields induce human cryptochrome to modulate intracellular reactive oxygen species. Plospiol. 2018. 16. E2,006,229. Cross, Reef. 261. Ahmad M. Niesner C. Gehring D. Wilczko W. Light-dependent magnetoreception in birds. The crucial step occurs in the dark. J. R. Sock. Interface 2016. 13. 20,151,010. Cross. 262. H.J. Biskup T. Katnig Dr. Hoare P. Alternative radical pairs for cryptochrome-based magnetoreception. J. R. Sock. Interface 2014. 11. 20,131,063. Cross Reef. 263. Procopio M. Ritz, T-radical pair-based avian magnetoreception. APS 2014 J10. 009. 264. Hogben H.J. Efimova. O. Wagner Rundell. N. Timmel. CR. Hoare. P. Possible involvement of superoxide and dioxygen with cryptochrome in avian magnetoreception. Origin of Zeeman resonances observed by in vivo EPR spectroscopy. Chem. Phys. Let. 2009. 480, 118 to 122. Cross Reefer. 265. Worcester S. Katnig. Dr. Hoare. P-spin relaxation of radicals in cryptochrome and its role in avian magnetoreception. JChem. Phys. 2016. 145. 035,104. Crossref. PubMed. 266. Katnig Dr. SOA, J.K., Solovyov, I.A., Hor p-electron spin relaxation can enhance the performance of a cryptochrome-based magnetic compass sensor. New J-Phys, 2016, 18, 063007. Cross-Reef, Quantum Rep, 2021, 3, 44. 267. Katnig Dr. Solovyov, I.A. Hor p-electron spin relaxation in cryptochrome-based magnetoreception. Phys. Chem. Phys. March 18, 2016, 12,443 to 12,456. Cross-Reef. PubMed, 268. Karogadinatai, Dranov, I.G., Sergieva, S.V., Stas, D.V., Steiner, UE kinetic magnetic field effect involving the small biologically relevant inorganic radicals NO and O2. ChemPhysChem 2011-12, 1714 to 1728. Crossref. 269. Urig G's exact results on dynamical decoupling by pi pulses in quantum information processes. New J-Phys 2008, 10, ERO 83024. Crossref. 270. Keynes R.H., Bedkihal, S., Katnig, Dr. Magnetosensitivity in dipolarly coupled 3-spin systems. Phys. Reverend Lett. 2018, 121, ISIO 96001. Cross-Ref. PubMed. 271. Babcock, Ens, Katnig, Dr. Electron-electron dipolar interaction poses a challenge to the radical pair mechanism of magnetoreception. J-Phys. Chem. Lett. 2020, 11, 2414 to 2421. Crossref. 272. Katnig, Dr. Radical pair-based magnetoreception amplified by radical scavenging. Resilience to spin relaxation. J-Phys. Chem. B. 2017, 121, 10,215 to 10,227. Cross-Reef. 273. Katnig Dr. Hor, P. The sensitivity of a radical pair compass magnetoreceptor can be significantly amplified by radical scavengers. 8. Rep. 2017, 7, 1 to 12. Crossref. PubMed. Endres S., Granzin J., Circalone F., Stadler A., Kraus U., Dreper T., Svensson V., Knieps Grunegan, E. Wirtz A., cousin A structure and function of a short love protein from the marine phototrophic bacterium Dinorosiobacter shebai. BMC Microbial. 2015-15, 1 to 16. Crossref. PubMed. 275. Mancia F., Smith G., Evans P. Crystal structure of substrate complexes of methylmalonyl-CoA mutase. Biochemistry 1999, 38, 7999 to 8005. Cross-Reef. PubMed. Tollinger M., Conrad, R., Hilbert, B.H., Marsh, Eng, Krautler. B. How a protein prepares for B12 binding, structure and dynamics of the B12 binding subunit of glutamate mutase from Clostridium tetanomorphum. Structure 1998, 6, 1021 to 1033. Cross-Ref. 277. Bender GN, Poyner, RR, Reed, GH identification of the substrate radical intermediate derived from ethanolamine during catalysis by ethanolamine ammonia lyase. Biochemistry 2008, 47, 11,360 to 11,366. Cross-Ref. Cross-Reef. Efimova, O. Hor, P. Role of exchange and dipolar interactions in the radical pair model of the avian magnetic compass. Biophys. J. 2008, 94th, 1565 to 1574. Cross-Reef. Nor D. Paulus B., Rodriguez R., Okafuji A., Bittel R., Schleicher E., Weber S. Determination of radical distances in light-active proteins and their implication for biological magnetoreception. Anju, Chem. Int ed. 2017, 56, 8550 to 8554. Cross-Reeve, 279, 280. Nordi, Franz S., Rodriguez R., Paulus B. Essen L.O., Weber S., Schleicher E. Extended electron transfer in animal cryptochromes mediated by a tetrad of aromatic amino acids. Biophies. J. 2016, 111, 301 to 311. Cross-Ref. Cross-Ref. Zoltowski B.D., Vaidya at Top D, Wydom J., Young M.W., Crane B.R. structure of full-length Drosophila cryptochrome. Nature 2011, 480, 396 to 399. Cross-Ref. 282. Levy C., Zoltowski B.D., Jones R., Vaidya, at Top D, Widom J., Young M.W., Scruton N's, Crane Bar, Lees D. Updated structure of Drosophila cryptochrome. Nature 2013, 495, E3E4. Cross-Ref. 283. Muller P., Yamamoto J., Martin R., EY S., Brettel K. Discovery and functional analysis of a fourth electron-transferring tryptophan, conserved exclusively in animal cryptochromes, and 6-4, photolysis, Chem. Common. 2015, 51, 15,500, and 2 to 15,505. Cross-Reef. PubMed. 284. Chagavets, M. Grissom, CB magnetic field effects in adenosyl-CoB 3. Alamine photolysis, relevance to B12 enzymes. J. Am Chem. ESOC, 1993, 115, 12,152 to 12,157. Cross-Ref. 285. Jones R., Hay S., Woodward J.R., Scruton NS magnetic field effect studies indicate reduced geminate recombination of the radical pair in substrate-bound adenosyl-cobalamin-dependent ethanolamine ammonia liase. J. Am Chem. SOK 2007, 129, 15,718 to 15,727. Cross-Ref. 286. Henbest KB, Maeda K., Hoare P., Joshi M., Bacher A., Bittel R., Weber S., Timmel CR, Schleicher E. magnetic field effect on the photoactivation reaction of Escherichia coli DNA photolysis. Proconnatal ACAD, AD. USA 2008, 105, 14,395 to 14,399. Cross-Ref. 287. Zhang M., Wang L., Zhong D. Photolyase, Dynamics and Electron Transfer Mechanisms of DNA Repair. Arch. Biochem. Biophies. 2017, 632, 158 to 174. Cross-Ref. 288. Hoare P., a DNA-based magnetic sensor. ACS Publications, Washington, D.C., USA, 2018. 289. Zhuang T.J., T.H., Zhong D., Barton J.K. A compass at weak magnetic fields using thymine dimer repair. ACS Cent. Sci. 2018, 405 to 412. Cross-Ref. 290. Masiha H.L., Wongnate T., Chayan P., Jones R., Scruton NS magnetic field effects as a result of the radical pair mechanism are unlikely in redox enzymes. J.R. Sock. Interface 2015-12, 20,141,155. Crossref. 291. Ritz T., Thalau P., Phillips J.B., Wilczko R., Wilczko W. Resonance effects indicate a radical pair mechanism for avian magnetic compass. Nature 2004, 429, 177 to 180. Crossref. 292. Engels S., Schneider N.L., Lefelt N., Heinz M., Zapka M., Michalik A., Elbers D., Kittel A., Hoare P., Moritzen H. Anthropogenic electromagnetic noise disrupts magnetic compass orientation in a migratory bird. Nature 2014, 509, 353 to 356. Crossref. 293. Hiscock H.G., Moritzen H., Manolopoulos Day, or P. Disruption of magnetic compass orientation in migratory birds by radiofrequency electromagnetic fields. Biophys. J. 2017, 113, 1475 to 1484. Crossref. Quantum Rep. 2021, 345. 294. Cavokin. K. The puzzle of magnetic resonance effect on the magnetic compass of migratory birds. Bioelectroman. J. Bioelectroman. ESOC, SOC, Phys. Regal, Biol, Med, EA, Bioelectroman ASOC, 2009, 30, 402 to 410. Crossref. PubMed, 295. Hiscock HG, Worcester S, Katnig Dr, Steers C, Jin Y, Manolopoulos Day, Moritzen H, Hor P. The quantum needle of the avian magnetic compass. Proconnatal Acad. AD. USA 2016, 113, 4634 to 4639. Cross Reef. Pub Med. 296. Harbour Simp. Quant. Bile. 1953, 18. Watson J. Crick F. The structure of DNA. Cold Spring 123 to 131. Crossref. PubMed. 297. Patel. Ad Grover's algorithm in natural settings. Archive 2020. Archive 2001. 00214. 298. Marletto. See Constructor Theory of Life. J.R. Sock. Interface 2015-12. 20,141,226. 299. Chatterjee N. Walker. GC Mechanisms of DNA Damage, Repair and Mutagenesis. Environ. Moll. Mutagenes. 2017 58. 235 to 263. Cross-Ref. 300. Wang W. Hellinger. H.W. Bies L.S. Structural Evidence for the Rare Tortimer Hypothesis of Spontaneous Mutagenesis. Proconnatal Acad. AD. USA 2011, 108, 17,644 to 17,648. Crossreef. 301. Bebenek K. Pedersen, L.C. Kunkel TA Replication Infidelity via a mismatch with Watson-Crick geometry. Proc. Natal Acad. AD. USA 2011. 108, 1,862 to 1,867. Crossref. 302. Coag, M.C., Lee. S. Insights into the effect of minor groove interactions and metal cofactors on mutagenic replication by human DNA polymerase beta.
Biochem. J. 2018, 475, 571 to 585. Crossref. 303. Kimsey I.J. Petzold K. Sathyamurthy B. Stein Z.W. Al-Hashimi H.M. Visualizing transient Watson-Crick-like mispairs in DNA and RNA duplexes. Nature 2015, 519, 315 to 320. Cross-Reef 304. Kimsey I.J. Szymanski S. Zahorancik W.J. Shakya A. Zhu Y. Chu C.C. Sathyamurthy B. Su Z. Al-Hashimi H.M. Dynamic basis for DG. DT misincorporation via tautomerization and ionization. Nature 2018, 554, 195 to 201. Cross-Reef. PubMed. Reverend Mod. Phys. 1963, 35, 724. Cross-Reef. 306. Mullush. Spenkelink. LM. Van Oygen. And when proteins play tag, the dynamic nature of the replisome. Biophys. Reverend 2019, 11, 641 to 651. Crossref. 307. Tolosa S. Sanson J. Hidalgo. Mechanisms for guanine cytosine tautomeric equilibrium in solution via steered molecular dynamic simulations. J. Moll. Leak. 2018, 251, 308 to 316. Crossreef. 308. Tolosa S. Sanson J. Hidalgo. Mechanisms of the TA to CG transition studied by SMD simulations, deamination versus tautomerization.
J. Mol leak, 2020, 308, 113,036. Cross Reef, 309. Li P. Rangadorai, A. Al-Hashimi H.M. Hams-Schiffer, S. Environmental effects on guanine-thymine mispair tautomerization explored with quantum mechanical, molecular mechanical free energy simulations. J. Am Chem. ESOC, 2020, 142, 11,183 to 11,191. Crossref. PubMed 110. SCL, Florian. JDNA polymerase, lambda active site, favors a mutagenic mispair between the enol form of deoxyguanosine triphosphate substrate and the keto form of thymidine template, a free energy perturbation study. JPhys. Chem. B 2017, 121, 7,813 to 7,822. Crossreef. 311. Rangadurai A. Simansky. S. Kimsey. I-Shi H. Al-Hashimi H.M. Probing conformational transitions towards mutagenic Watson-Crick-like G. Mismatches using off-resonance sugar carbon, 1 Randa relaxation dispersion. J. Biomol. NMR 2020, 74, 457 to 471. Cross Reef. 312. Burke K. Perspective on density functional theory. J. Chem. Phys. 2012, 136, 150,901. Crossref. 313. Kramer. C.J. Essentials of Computational Chemistry. Theories and Models. John Wiley and Sons, Hoboken, N.J., USA, 2013. 314. Jensen. F. Introduction to Computational Chemistry. John Wiley and Sons, Hoboken, N.J., USA, 315. Broveritz, OHO, Hoveran DM proton, tunneling in the A. Watson-Crick DNA base pair, myth or reality? J. Biomol. Struct. 2015, 33. 2,716 to 2,720. Crossref. Broveritz, OHO, Super, Kays, Hoveran, DM novel pathway for mutagenic tautomerization of classical A. DNA base pairs via sequential proton transfer through quasi-orthogonal transition states, a QM-Q-time investigation. PLS 1 2018, 13. ESRO 100 and 99,044. Crossref. 317. Broveritz, OHO, Hoveran, DM, how many tautomerization pathways connect Watson-Crick-like G? DNA base mispair and wobble mismatches. J. Biomol. Struct. Gin 2015, 33, 2,297 to 2,315. Crossref.
Crossref.
Broveritz, OHO, Super, Kays, Dynets, A. Hoveran, DM unexpected roots of the mutagenic tautomerization of the T-nucleobase in the classical A. DNA base pairs, a QM-Q-time comprehensive view. Front. Chem. 2018, 6, 532. Crossref. PubMed. 319. Broveritz, OHO, Hoveran, DM tautomization of the A. Watson-Crick base pair. Via double proton transfer provoke point mutations during DNA replication. A comprehensive QM and Q-time analysis. J. Biomol. Struct. DIN 2014, 32, 127 to 154. Crossref. PubMed. Broveritz, OHO, Hoveran, DM. Why the tautomerization of the G? C. Watson-Crick base pair via the DPT does not cause point mutations during DNA replication. QM and Q-tame comprehensive analysis. J. Biomol. Struct. DIN 2014, 32, 1,474 to 1,499. Crossref. PubMed. 321. Villani G. Theoretical investigation of the coupling between hydrogen atom transfer and stacking interaction in adenine-thymine dimers. Chem Physchem 2013, 14, 1,256 to 1,263. Cross-Reef. 322. Solopolo D, Mendieta Moreno G, Trabada DG, Mendieta J, Ortega J proton transfer in guanine cytosine base pairs in bDNA. J-Chem. Theory Comput. 2019-15, 6,984 to 6,991. Crossref. PubMed, Quantum Rep. 2021, 346. 323. Rosbach S. Oxenfeld. See influence of coupling and embedding schemes on QM size convergence in QMMMM, approaches for the example of a proton transfer in DNA. J-Chem. Theory Comput. 2017-13 1100 and 2 to 1107. Crossref. 324. Broveretz. Oho, Hoveren. DM atomistic mechanisms of the double proton transfer in the H-bonded nuclear base pairs. QMQ time computational lessons. J. Biomol. Struct. DIN 2019. 37. 1880 to 1907. Cross-Reef. 325. Jarevka P. Spana J. Cerny J. Hobza P. Benchmark database of accurate. MP2 and CCSD-T, complete basis set limit, interaction energies of small model complexes, DNA base pairs, and amino acid pairs. Phys. Chem Shem. Phys. 2006, 8, 1985 to 1993. Crossref. 326. Van der Wicht, T. Gera, CF, Swart M. Bickelhaupt FM performance of various density functionals for the hydrogen bonds in DNA base pairs. Chem. Phys. Let. 2006, 426, 415 to 421. Cross-Ref. 327. Bende. A hydrogen bonding in the urea dimers and adenine thymine DNA base pair, anharmonic effects in the intermolecular H bond and intramolecular H stretching vibrations. Fior. Chem. ACC. 2010, 125, 253 to 268. Cross-Riff. 328. Godbeer. A quantum tunneling effect in DNA base pair mutation. University of Surrey, Guildford, UK, 2014. 329. Godbeer A. Al-Khalili. J. Stevenson P. Environment-induced dephasing versus von Neumann measurements in proton tunneling. Phys. Reverend a 2014. 90. 012,102. 330th. Godbeer A. Al-Khalili. J. Stevenson P. Modeling proton tunneling in the adenine thymine base pair. Phys. Chem. Phys. 2015, 17, 13,034 to 13,044.
Cross-Ref.
PubMed. 331. Pol R. Soccer O. Slavivchek P. Sala M. Hodgkinson P. Dravchinsky M. Proton transfer in guanine cytosine base pair analogues studied by NMR spectroscopy and PEMD simulations. Faraday discuss. 2018, 212, 331 to 344. Crossref. 332. Fang W, Chen J, Rossi M, Feng E, Li, 10th C. Michaelides A. Inverse temperature dependence of nuclear quantum effects in DNA base pairs. J. Phys. Chem. Let. 2016, 7, 2,125 to 2,131. Cross-Reef. 334. Clegg, R.M. The History of Fret. In Reviews in Fluorescence 2006. Springer, Berlin, Heidelberg, Germany. 335. Kenkray, V. Knox, R. Theory of Fast and Slow Excitation Transfer Rates. Phys. Reverend Let, 1974, 33, 803. Crossref. 336. Forster, T. Delocalized Excitation and Excitation Transfer. Sinanoglu O. ed. Modern Quantum Chemistry. Istanbul Lectures 3. Academic Press. New York, NY, USA, London, UK, 1965. 337. Chinelli Ra, Tozzini, F. Pellegrini the First, Beltram F. Cerullo G. Zavellani Rossi M. De Silvestri S. Tiaghi M. Giacca M. Coherent Dynamics of Photoexcited Green Fluorescent Proteins. Phys. Reverend Lett. 2001. 86. 3439. Cross, Ref. 338. Zhangji, Ma, Y, Prawl, B.S. Fleming GR ultrafast fluorescence depolarization in the yellow fluorescent protein due to its dimerization. Chem Phys Chem 2005, 6, 1628 to 1632. Cross, Reef, 339. Shi, S. Kumar P. Lee KF generation of photonic entanglement in green fluorescent proteins. Nat. Common. 2017, 8, 1-7. Cross Reef. 340. Kim E. Paul III HL, Chen A. Tamorphalau Chiich, Nwinta, Kleiger, D.S. blank P's, Vogel SS Venusat 206 dimers behave coherently at room temperature. Biophys. J 2019, 116, 1918 to 1930. Cross Reef.
Bud. 141.
Dietrich C.P., Stude A., Tropf L., Schubert M., Cronenberg N.M., Osterman K., Hoffling S., Gather MC and Exciton polariton laser based on biologically produced fluorescent protein. SCI. Aid V. 2016, 2, E1 million. 600,666. Cross Reef. PubMed, Sanchez Mostiro, Koopman M., Van Dyke M., Hernando J., Van Holst NF, Garcia Parajo MF photon antibunching proves emission from a single subunit in the autofluorescent protein DS red. ChemPhys Chem 2004, 5, 1782 to 1785. Cross Ref. PubMed 343. Visser NV. Hink. Ma. Borst. JW. Van der Crocht GN. Visser. AJ circular dichroism spectroscopy of fluorescent proteins. Thebs Let. 2002, 521, 31-35. Cross Reef. 344. Kushik SV, blank, PS, Vogel, SS anomalous surplus energy transfer observed with multiple FRET acceptors. PLOS 1 2009, 4, E8031. Cross Reef. 345. Lunas B, Dice J, Roselle, F, Boxer SG, Mona W. Photophysics of DS Red, a red fluorescent protein, from the ensemble to the single molecule level. J. Phys. Chem B 2001, 105, 5048 to 5054. Cross Reef. 346. Shi S. Thomas A., Kordzo N.V., Kumar P., Huang E., Lee K.F. broadband photon pair generation in green fluorescent proteins through spontaneous four-wave mixing. SCI, Rep. 2016, 6, 24,344. Cross-Ref. 347. Xian Rai, the green fluorescent protein. Anu, Reverend Biochem, 1998, 67, 509 to 544. Cross-Ref. 348. Rodriguez E.A., Campbell Ri, Lin Ji, Lin Imzi, Miyawaki A., Palmer A., Shu X., Zhang J., Tien Rai, the growing and glowing toolbox of fluorescent and photoactive proteins. Trends Biochem. Psi, 2017, 42, 111 to 129. Cross Reef. 349. Gross L.A., Baird G's, Hoffman RC, Baldridge KK, Tien Rai, the structure of the chromophore within DS Red, a red fluorescent protein from coral. Proc. Natal Akkad. Psi. USA 2097, 11,990 to 11,995. Cross Reef. PubMed. 350. Shana NC, Lambert GG, Chamus A, Ni Y, Cranfill PJ, Baird Ma, Cell BR, Allen JR, DRN, Israelson M, a bright monomeric green fluorescent protein derived from Branchiostoma lanceolatum. Nat. Methods 2013-10, 407 to 409. Cross-ref. PubMed 351. Ward W. Cormier M. Energy Transfer Protein in Coelenterate Bioluminescence. J. Biol. Chem. 1979, 254, 781 to 788. 352. Arpino Jha. Rizkala. P.J. Jones D&D crystal structure of enhanced green fluorescent protein to 1.35. A resolution reveals alternative conformations for glue 222. PLOS 1 2012. 7. E47132. Cross Reef. 353. Tagizada R.R. Shirley, JLCFP and YFP, but not GFP, provides stable fluorescent marking of rat hepatic adult stem cells. J. Biomed. Biotechnol. 2008. 2008. 453,590. Cross-ref. Quantum Rep. 2021. 347. 354. Chudakov DM. Lukyanov S. Lukyanov car-fluorescent proteins as a toolkit for in vivo imaging. Trends Biotechnol. 2005. 23. 605 to 613. Cross-ref. 355. Hebish E. Nebel J. Landsberg J. Frey E. Leisner M. High variation of fluorescence protein maturation times in closely related Escherichia coli strains. PLS1 2013. 8. S75991. Gather, SMC Yun, Shabio-optimized energy transfer in densely packed fluorescent protein enables near-maximal luminescence and solid-state lasers. Nat. Common. 2014. 5. 1 to 8. Cross Reef. 357. Spect EA. Brazelman. E. Palmer AA, a critical and comparative review of fluorescent tools for live cell imaging. Anu. Reverend Physial. 2017. 79. 93 to 117. Cross-ref. 358. Beleza E. Kim. J.M. Clusal P. Systematic characterization of maturation time of fluorescent proteins in living cells. Nat. Methods 2018, 15, 47 to 51. Cross-reef. 359. Sarkar P. Kushik. S.V. Vogel. S.S. Grichinsky 1. Grichinsky ZK photophysical properties of cerulean and venous fluorescent proteins. J. Biomed. Opt. 2009. 14. 034,047. Cross-ref. PubMed. 360. Gilmore J. Mackenzie. RH spin boson models for quantum decoherence of electronic excitations of biomolecules and quantum dots in a solvent. J. Fis. Condons. Matter 2005, 1735. Cross-ref. 361. Godhart J. Von Stetten D. Noirclerc Savoy M. Lellemazin M. Jewson L. Hink Ma. Van Weeren L. Gadella 2, Royant a structure-guided evolution of cyan fluorescent proteins towards a quantum yield of 93%. Nat. Common. 2012, 3, 1 to 9. Cross-reef. PubMed 362. Alga W.A. Hildebrandt N. Vogel S.S. Medinz. Ilfret as a biomolecular research tool, understanding its potential while avoiding pitfalls. Nat. Methods 2019. 16, 815 to 829. Cross-reef. 363. Siegel R.M., Chan, F.K.M., Zacharias Da, Swafford R., Holmes K.L., Tsian, Rai, Leonardo. MJ measurement of molecular interactions in living cells by fluorescence resonance energy transfer between variants of the green fluorescent protein. S.I. Signal. 2000. Plone. Cross-reef. 364. Stryer L. Haugland, RP energy transfer, a spectroscopic ruler. Proc. Natal Aked SEI. USA 1967, 58 719. Cross-reef. 365th. Gilmore, J. Mackenzie. RH Quantum Dynamics of Electronic Excitations in Biomolecular Chromophores, Role of the Protein Environment and Solvent. J. Phys. Chem. A 2008, 112, 2162 to 2176. Crossref. 366. Bors J.W., Hinkmar, Van Hoek, A. Visser A.J. Effects of refractive index and viscosity on fluorescence and anisotropy decays of enhanced cyan and yellow fluorescent proteins. J. Floresque, 2005, 15, 153 to 160. Crossref. 367. Geddes C.D., Lakowicz, J.R. Reviews in Fluorescence 2006, Springer, Berlin, Heidelberg, Germany, 2005. 368. Barova N. Dibari, L. Pesatelli G. Application of electronic circular dichroism in configurational and conformational analysis of organic compounds. Chem. Soc. Reverend 2007, 36, 914 to 931. Crossref. 369. Grishina, I.B., Woody, R.W. Contributions of tryptophan side chains to the circular dichroism of globular proteins, exciton couplets and coupled oscillators. Faraday Discuss. 1994, 99, 245 to 262. Cross Reef. 370. Illigan, R.P., Rhodes E., Gruber D.F., Cow, H.T., Pieriboneva, Regan L., a new bright green-emitting fluorescent protein-engineered monomeric and dimeric forms. Febs, J. 2010, 277, 1967 to 1978. Cross Reef. Stewart H. Quantum Computation in Brain Microtubules. The Penrose-Hameroff, Orch or Model of Consciousness. Phylos, Trans, R. Soc. Lund, Sir, A Math. Phys. Eng Tsai. 1998, 356, 1869 to 1896. Cross Reeve. 372. Penrose R. Hameroff, S. Consciousness in the Universe. Neuroscience, Quantum Space-Time Geometry, and Orch or Theory. J. Cosmol, 2011, 14, 1-17. 373. Hameroff, Senior, Penrose R. Consciousness in the Universe, an updated review of the Orch or Theory. In Biophysics of Consciousness, a foundational approach. World Scientific Publishing Company, Pte. Limited, Singapore, 2016, pp 517 to 599. 374. Bezanilla, F. Voltage-gated ion channels. In Biological Membrane Ion Channels, Springer, Berlin, Heidelberg, Germany, 2007, pp 81 to 118. 375. Vaziri A. Plenio. MB Quantum Coherence in Ion Channels, Resonances, Transport and Verification. New J. Phys. 2010, 12, 085001. Cross Reeve. 376. Doyle Da, Cabral J.M., Futzner Ra Quo A. Gulbis J.M., Cohen, S.L., Chait, B.T., McKinnon, are the structure of the potassium channel, molecular basis of Kp-Lus conduction and selectivity. Science, 1998, 280, 69-77. Cross-Ref. 377 are potassium channels and the atomic basis of selective ion conduction. Nobel Lecture, Anju, Chem, Int. ed. 2004, 43, 4265 to 4277. Cross Reef. 378. CO. S.A. Sig. D. Papazian, D.M. Bezanilla, F. Voltage sensing residues in the S2 and S4 segments of the Shaker K+ channel. Neuron 1996, 16, 1159 to 1167. Crossref. 379. Alan Tukuyukak, S. Chung, S.H. Molecular Dynamics Study of the KCSA Potassium Channel. Biophys. J. 1999, 77, 2502 to 2516. 380. Baker K.A., Chitzalonis C., Kwiatkowski W., Cho S. Riek R. Conformational dynamics of the KCS. A potassium channel governs gating properties. Nat. Struct. Mol Bile. 2007, 14 1089 to 1095. Cross Reef. 381. Chung S.H. Alan 2 Kuyukak S. Conducting state properties of the KCSA potassium channel from molecular and Brownian dynamics simulations. Biophys. J. 2002, 82, 628 to 645. Cross Ref. 382. Ruby. Shulton K. Computational Studies of Membrane Channels. Structure 2004, 12, 1343 to 1351. Cross-ref. PubMed 383. Sumhammer, J. Suliok, G. Bernreuter G. Quantum dynamics and non-local effects behind ion transition states during permeation in membrane channel proteins. Entropy 2018, 20, 558. Cross-reef. PubMed, Quantum Rep. 2021, 348. 384. FISEC Z. Swain. S. Quantum Interference and Coherence. Theory and Experiments. Springer Science and Business Media. Berlin, Germany. 2005. Volume 100. 385th. Solari 5th. Moradi N. Sajadi M. Fazila F. Shabazi F. Quantum decoherence timescales for ionic superposition states in ion channels. Phys. Reverend E. 2015 91st, 032704. Crossref. PubMed. Ganem Z. Tokmekov. A. Viziria vibrational excitons in ionophores experimental probes for quantum coherence, assisted ion transport and selectivity in ion channels. New J. Phys. 2011, 13, 113,030. Crossref. 387. Bernreuter G. Sumhammer J. Can quantum entanglement between ion transition states effect action potential initiation. Kong Comput. 2012 4, 29 to 37. Crossref. 388. Coswell. Ab quantum electrochemical equilibrium, quantum version of the Goldman-Hodgkin-Katz equation. Quantum Rep. 2020, 217. Crossref. 389. Barjas Caswell, A. Quantum tunneling of ions through the closed voltage-gated channels of the biological membrane, a mathematical model and implications. Quantum Rep. 2019, 119. Cross Reef. 390. Caswell, Ab lithium stabilizes the mood of bipolar patients by depolarizing the neuronal membrane via quantum tunneling through the sodium channels. Clin. Psychopharmacol. Neurosia. 2020, 18, 214. Cross Reef. 391. Al-Rabia M. Caswell, Ab. Suleiman, A. Krisha L. Role of potassium ions quantum tunneling in the pathophysiology of phantom limb pain. Brainsci. 2020-10, 241. Cross Reef. 392. Wen L. Zhang X. Tian Wei. Zhang L. Quantum confined superfluid from nature to artificial. Sci. China Marta. 2018, 61, 1027 to 1032. Cross-ref. Quantum confined ion superfluid in nerve signal transmission. NanoRes. 2019-12, 1219 to 1221. Cross-reef. 394. Froelich F. McCormick. DA endogenous electric fields may guide neocortical network activity. Neuron 2010, 67, 129 to 143. Cross-ref. 395. Shivacharan, Rs, Zhang M. Durand DM. Can neural activity propagate by endogenous electrical field? J. Neurosi. 2015, 35th, 15,800 to 15,811. Cross-ref. Francis J. T. Gluckman, B.J. Schiff, S.J. Sensitivity of neurons to weak electric fields. J. Neurosi. March 23, 2003, 7,255 to 7,261. Cross-ref. 397. Jedlicka P. Revisiting the Quantum Brain Hypothesis Toward Quantum Neurobiology. Front, Moll, Neurosi. 2017-10, 366. Cross-ref. 399. Anastasiou, Say, Koch. See ephaptic coupling to endogenous electric field activity. Why bother? Kerr, Opan, Neurobile, 2015, 31, 95 to 103. Cross-ref.